of environmental factors then interact with all of these sort of aforementioned factors there. So there's a couple of common types of population structures that we typically encounter, right? In the simplest form, we have what we call sort of a panmictic population structure, where generally there is global density dependence, and the individuals are sort of genetically homogeneous. We also have spatial heterogeneity, where again, this is a global density dependence, but there's a potentially spatial heterogeneous abundance across space. In addition, we might have a metapopulation structure where essentially each region has its own sort of stock recruitment function. And we typically, in this case, assume that environmental drivers or factors determine sort of the demographic rates. And the last somewhat common population structure we typically encounter is natal homing, where again, we have local density dependence, and the natal origin or genetic factors will typically determine demographic rates. And so for tunas, it kind of encompasses all of these sort of population structures that I determined. So perhaps for some of our next gen assessments, these structures might be the forefront of what we think about incorporating when we start developing a model. So there's a couple of several data sources that can be used to infer or estimate spatial structure. So we have genetics, where for sort of bluefin, we have CKMR or SNPs, maybe to tell apart population structure. And in some cases, we might have stock composition data, such as otolith microchemistry, to help inform sort of the stock or population proportions. And so for next gen assessments, it might be prudent to think about how we might incorporate these submodels in. In terms of fishery data sources, we have our traditional sort of composition catch, as well as indices, and typically comps only comes in the form of length composition data. We also have tagging data, so this can come in the form of archival satellite tags, acoustic tags, or FLOI conventional tags. So it might be necessary for next gen assessments to be able to incorporate the full sort of suite of these tagging types. And the last somewhat unique, I guess, data source is sort of environmental fields, which is typically used in some of our more fine scale spatial models to help drive movement or recruitment dynamics there. So there's a couple ways we can model either implicitly or explicitly spatial structure. But I think in the most general form, most of our population dynamics should try to incorporate maybe different populations as well as different regions and different time steps, so yearly or sort of quarterly or seasons, ages and or lengths, as well as sexes as well. And I think most of our spatial structure, at least right now, is typically represented with what we call like spatially stratified or large box approaches. But there's been increasing use of fine scale population dynamics models like SPM or CPDIM that I'll touch briefly on, but I'll sort of punt that over to the other speakers later. So in order to sort of model spatial structure, we can potentially implicitly represent spatial structure with what we call as panmictic approximations, right? So for some tuna assessments, fleets as areas models are relatively common, where we essentially treat fishery fleets as different areas, and we try to use selectivity to approximate spatial structure in sort of the age or length composition. So that's sort of one way we might be able to approximate spatial structure implicitly. The other sort of newer approach here is this numbers at age process error, where essentially we have an exponential mortality model, and then we have process error from the exponential mortality model. And so here it implicitly accounts for unexplained movement dynamics, but also critically unexplained mortality processes as well. But there are a lot of caveats with this method, where these process errors are really only as good as the signal in your data. And I think it's pretty important to try to understand why certain patterns emerge rather than, blindly trusting the modeled process error there. The sort of last panmictic approximation approach I'll talk about is pretty simple. Essentially, we can approximate spatial structure by conducting independent assessments in each region. And so here you might treat region 1 as one assessment, region 2 as one assessment, and region 3 as another assessment, assuming no movement. And I think maybe in these cases, numbers at age process error can be helpful in accounting for sort of the lack of movement modeled in these models. The other way to approximate spatial structure is what we call these spatially stratified approximations, where essentially we're treating large spatial strata as a single area that we model. And so generally these spatial strata are defined based on management boundaries of how catch is being reported. And it implicitly assumes that dynamics within each strata are sort of homogeneous. And so there are also some models that uniquely define sort of a hybrid, what I'll call a hybrid model, where there's a large box spatial strata defined, but then within that strata, they'll additionally define sort of a Fleets' areas approach as well. So in this example, essentially 1N, 1C, and 1S is considered a single spatial strata, and then the N, C, and S are separate fleets within that strata. And so perhaps maybe for some of our next-gen models, that might be certain features we might think about incorporating. And some of these spatially stratified approximations range sort of a variety of a number of spatial strata modeled, right? We can range from up to sort of 2 areas modeled as well as 10 areas modeled, sort of all the way down here. And some assessments also include population-specific dynamics. So what they call stocks is essentially they have 2 or 3 sort of unique populations modeled in addition to their regions. And sort of the other way we can approximate spatial structure is more recently, at least, these fine scale spatial approximations. I'll touch briefly on this, but essentially, population dynamics are defined on really fine spatial strata. So about, it can range from about 50 to 200 spatial small strata. And within each spatial strata, then you'll be modeling recruitment, mortality, and movement processes as well. Generally, so movement rates are going to be defined for all spatial strata, and you would expect maybe a bunch of parameters to be estimated. So typically, estimation is constrained using preference functions as informed by sort of these fine scale environmental fields to drive population dynamics as well as movement dynamics. And some of these models, I think, generally require georeference data on both catch and or effort to help inform fishing mortality across space. And so right now, a lot of these models are used for research and operating models rather than for providing management advice. So the last sort of theoretical framework or approximation I'll talk briefly about is multi-scalar frameworks. So I think there hasn't been that many examples, and I'm not as familiar with this, but essentially you have a fine scale sort of tagging submodel, and you can then aggregate movement estimates from the fine scale tagging submodel to match up with the scale of the coarse spatial strata, and then your population dynamics operate on this coarse spatial strata here. And so I think there's 2 ways to potentially approach this, but there's some practical constraints. We can do it in this integrated analysis framework where we integrate a fine scale spatial tagging model with the coarse scale population dynamics models. but I think you'll likely run into runtime and over-parameterization constraints. And so I think under this multi-scalar framework, it's likely that a two-stage approach is most likely the most tractable approach, where we have essentially two submodels, and you either use the movement estimates from the fine scale model as priors or known values and plug those into your coarse scale population dynamics models. So I'll sort of transition next into talking about some common decision points for spatial assessments that I think are generally applicable for all of the different approximations I talked about previously. So first and foremost is whether or not a spatial model is really needed, and whether the data support the use of a spatial model. I won't really touch on this since it's mostly we're focusing on parts of a spatial model we might decide to implement. And so I'll primarily focus on how or what recruitment dynamics look like, or considerations we might take into, whether or not movement varies across space and how that might vary across population partitions. How do we determine the initial scale by region, and how demography varies across space, and how we address changes in biology, and then how fleet structure should be represented. So in terms of recruitment dynamics, Andre sort of outlined this general model where here we have global density dependence. Essentially, we have global spawning stock biomass determines global recruitment, and then that gets apportioned out by a sex ratio, a spatial allocation that might be time varying or time invariant, and it might also be apportioned out by season as well. We also might have a local sort of density dependence where region-specific spawning stock biomass determines region-specific recruitment, and then that also gets apportioned out by sexes, by seasons, and by spatial sort of strata. And so some decision points here for spatial models in general is whether or not density dependence is global or local, and whether or not recruitment is seasonal or annual. Also, how recruitment might be apportioned spatially, so whether or not this is time varying or time invariant. And for recruitment deviations, whether or not deviations vary by time only or across time and space. I think another consideration we might think about is whether or not recruitment deviations are correlated across areas. So something like an AR1 or some sort of multivariate normal correlation. I don't know if there's been any examples of correlated process error in recruitment here, but I think maybe this is something we should also think about. So the sort of full gamut of recruitment types can likely be summarized with these three different models, where we have local density dependence with locally varying recruitment deviations and time varying deviations. We might also have global density dependence with a time invariant sort of allocation of recruitment spatially, but then have time varying recruitment devs, and then again global density dependence with time varying recruitment apportionment, and then also time varying recruitment deviations as well. So the next sort of topic or decision point I'll talk about is connectivity and movement and how we might model that. And I'm sure a lot of people have experienced attempting to model movement and realize that it can be particularly difficult, just given the sparse data that's available to inform movement, as well as the number of parameters to estimate them. And so if we do sort of a pairwise movement approach where we estimate movement parameters for every single area, the number of parameters will increase quadratically with the number of areas we're modeling. And this further compounds if we assume movement varies by, population, age or length, and sexes and seasons as well. So There are a couple of approaches that assessments have done to sort of simplify or facilitate movement estimation, and these might be considerations that we might consider when incorporating different movement parameterizations in some of our next gen models. So in the simplest case, we might assume there's no post-settlement movement following recruitment. We might also have options to implement a gravity model where essentially we're estimating parameters for the diagonal of a movement matrix. So all of the residency rates, and then we fix the emigration rates or assume they're equal given the estimated residency. The multi-fan-CL essentially estimates parameters only for the adjacent regions. And then, so essentially you define an adjacency matrix, and then you derive the non-adjacent rates based on matrix inversion. And then you can also use priors and penalties to constrain movement. So if movement varies across sort of these population partitions, some assessments also do parameter blocking across these attributes. We might also link movement rates to covariates, or also assume movement arises from some sort of stochastic process as well. So something like an IID or a 1D AR1 correlation. And then I think Jim will likely talk about this, and I think Tobias as well, but assuming also movement arises from a mechanistic process where you can essentially parameterize movement to arise as a continuous Markov chain, and then have movement be derived from mechanistic processes like advection, diffusion, and taxis, and potentially represent these time or age varying effects as splines. So there's some other considerations for movement. So order of operations, so whether or not mortality or movement occurs first, or whether or not movement or mortality occurs, or if we want continuous movement to occur. And then the other one is whether or not recruits are allowed to move. As we know, recruitment and movement can be confounded. And so if enforcing that recruits don't move can potentially help alleviate some of that confounding as well. So the other decision point is how we determine the initial scale of the population. I think this is particularly difficult to do in spatial models, and particularly for tunas, just given the lack of fishery independent surveys. And so typically we do scaling, or in tuna assessments, they do scaling factors that they derive by standardizing CPUE, and then summing the relative sort of spatial abundance by large spatial strata to obtain regional relative abundance, and then using the scaling factors to rescale the regional indices along with imposing a constant catchability across space. And so it's generally well established for how we do this in spatially stratified models, but I'm not sure how this might interact with some of the fine scale population models. And I think maybe if you standardize CPUE within these fine scale models, maybe that's already sort of implicitly resolved from that. So I'm almost to the end there. I'll touch on demography and then fleet structure. So in terms of demography, it can be a little challenging in spatial assessments. And one of the primary challenges is ensuring that fish don't shrink or unmature. So there's two pretty common ways to not necessarily address this complication, but two common ways we've addressed differences in demography across space within these assessments. And so one is to assume sort of what we call phenotypic variation, where demographic rates change as soon as movement happens. So if an individual starts in region 1, it takes on the demographic rates in region 2. And as an individual moves from region 1 to region 2, it takes on the new rates of region 2 without accounting for the fact that it was previously in region 1. The other way to do this is to assume sort of genetic variation, where demographics are determined by the natal sort of population and they don't change. And so this requires tracking the natal population. But none of these are really, I guess, satisfactory or quote unquote realistic. And so we might envision some different approaches for modeling demography across space, right? We might assume that changes in demographic rates are length-based processes, or sort of model growth via growth transition matrices, or explicitly track sort of mature or immature partitions. We might also determine spatially varying demographics based on sort of a weighted average of the natal area as well as the regions that a given cohort visits. But I think a lot of these different approaches have remained a pretty unexplored aspect of spatial assessments. So the last thing I'll talk about is fleet structure. So for tuna models, there's often a wide range of fleets that are defined, just given the multinational nature of these fisheries. And so selectivity for these fleets can potentially be extremely complex and exhibit these dome-shaped patterns. And in some cases, selectivity might also be shared or mirrored across fleets. So in this example here, the middle row, those two panels are likely two different fleets sharing selectivities there. And I won't really touch on this, but fleet structure in some of these assessments are typically or can be defined using this sort of regression tree approach to define heterogeneity or to identify heterogeneity in sort of the length comp data as well. So I kind of threw a lot at people, and so there's some take homes here that I'll sort of try to drive. So there's a lot of decision points, and incorporating spatial structure can be very complex. And I think in general, some of our next gen models should be able to try at least attempt to accommodate all of the features that are discussed in these two papers, as well as what I talked about previously. And I think importantly, be able to accommodate a diversity of data sources and readily adapt to sort of new emerging data sources. I think another important thing is to be able to represent movement in a mechanistic manner, such as to reduce the degree of parameterization. And then also potentially readily extend from single region models, fleets as areas models, and then multi-population, multi-region models. There are some sort of, at least what I think are unresolved issues, but maybe people will disagree, but I think tagging models are likely a necessary component of next gen models, but it's not really clear how these should be configured, right? Whether or not we use fine scale tagging models or large box sort of tagging models. And along the same lines, whether or not next gen models need these multi-scalar frameworks and how exactly that would be implemented in the relative merits compared to stratified or large sort of stratified approaches. I think that random effects via sort of Laplace approximation should be part of next gen features, but sort of in my experience, I'm not sure how tractable that is with the number of model partitions that are often modeled in these large scale spatial sort of tuna models. But I think in RTME, it should be pretty easy to switch between penalized likelihood and Laplace approximation. And so I think that's it for now. I have some discussion questions listed there, but yeah, happy to take any questions.
Okay, thanks a lot, Matt. We only have a little bit of time left, so we'll take maybe a question, but make sure everyone keeps their questions for the discussion period after the next two talks. Does anyone want to start off with a question? Don't see any hands raised, which is fine. So let's just move on to the next presentation and start the discussion later on. Thanks, Matt.
Thanks.
So the next presentation is Jim Thorson. Jim, can you share your screen?
Unmuting, you can hear me?
I can hear you, yes.
Great. And then, okay, shoot. wrong order. Can you see my screen now?
Yes, we can. Yeah, but it's
Great.
not presentation mode yet.
Did that,
It is
so is
now.
that, okay.
Okay, good. Okay.
Um, yeah, so thank you. I'm excited to participate and excited about all the, energy to think through spatial modeling and in across areas and agencies. And thanks to the organizers, Mark and Arnie and many others. Okay, also, yeah, Matt did a great job of summarizing many different dimensions of choices we have to make. And I guess I'm going hard the other kind of dogmatic direction and claiming a single hammer for kind of a wide set of problems. And so my title here is that practical scale free and generic approach to movement modeling, bridging from individual based to population scale dynamics. And basically the main message here is that thinking about movement involves learning some new math. So um, there's some pieces here that some people are familiar with and others maybe not, but I guess I think learning some new math is worth the effort. It's I'm going to be summarizing pieces that are covered in chapter 4 and 11 of the book that Casper Christensen and I recently wrote, and inspired by conversations with many of the people on the call. And then, yeah, some of the concepts are also covered in this paper on the left that Matt started to talk about. Yeah, and so uh, my claim is that we have this sort of set of goals with movement analysis that we want to think about movement in a way that conserves numbers. So we want to move animals around in a way that's sort of isolated from other growth or mortality processes. Um, yeah, we want a movement model where we can easily calculate habitat utilization as a stationary distributions. We want a movement process that we can, like for instance, in equilibrium conditions, map out the um, portions in different strata. We want identical parameters in individual based, like Lagrangian and gridded Eulerian context. So this is sort of similar to Matt's claim about this sort of multi-scale, but maybe taking it a step further that you can, in fact, ideally run a model on individual track, not in discrete space, but in continuous space, and then use those same parameters in a gridded space model at a different scale. We want the model to be parsimonious and flexible. So using, as Matt again said, using covariates to try to collapse down a big unstructured movement model to a small set of parameters that we can estimate depending on how much data we have. We want it to be computationally efficient. We want the parameters to be scale free, so it automatically corrects for discretization choices. And I think we also want it to be continuous time. So, this can, well, often we want to have a model where we can back out, we can fit it at a certain time step, like annual or quarterly, but then we can back out what happened at times between those, um, within those intervals. So, um, my claim to do this, is that physicists have thought about this for a long time, and you know, we wouldn't know how to build an airplane or simulate fluid over simulate fluids without people having thought this through. And the way people do this is typically with these advection diffusion models. So um, the if you think about a density function in space and time, in continuous space and continuous time, the change at a given spot over time is explained in this version by sort of the curvature at a given spot, which is the diffusion, the spatial curvature. That's the the Lagrangian at a given spot. By a drift term, so you could have passive drift of larvae, for instance, in this kind of larval tracking. So you'd have a drift term and the gradient of your density and whether those point in the same direction or not. That's the second term. And then there's a taxus term. So you've got a preference function, the gradient of that um, defines in which direction animals will choose to advect in seeking out higher preferred habitat. So this PDE, you can take it and make it into an SPDE if you want to do the GMRF, the kind of spatiotemporal modeling. But for the most part, I'm just going to ignore stochasticity and discretize it in different ways to try to make the point. And I'm going to talk through different ways of discretizing this to try to convince people that it's not that complicated and that you can do it at different scales. So the decision tree that Casper and I end up with is this sort of ham-handed figure here, where again you've got this sort of vector field for oceanographic drift. Like Ken Anderson, I think, has this sort of global model that he's discretized and computed sort of, over a monthly time step and different depths. What's the exchange rate of different one spot to another based on global Earth systems models. There's also taxus, like a preference function that animals have that could be seasonal, so that forces them to move back and forth between spawning and feeding grounds, for instance. And then there's diffusion rates that explains the residual kind of unexplained stuff that's happening. Those go together in this partial differential equation. You then have to decide your model context, and that defines whether you want to treat it as continuous tracks on the bottom left, gridded high resolution tracks, which you can solve as a hidden Markov model, or these kind of gridded densities that you can fit with this matrix exponential. So I'll talk about the continuous tracks first. In 1D space, you might have, in these continuous space, you're treating the location of an animal as the variable of interest. And so an animal will have location S in time t, and it'll start at S0. And it has a bunch of, in this simplest case, if you if you equal intervals over time, you've got a bunch of these changes in location delta that have some unknown distribution G with a drift term. And if you sum a bunch of those together, it converges on a normal distribution under the central limit theorem. So you just take a bunch of equal intervals, sum them together, and you get a net change that's normally distributed with the variance times the number of intervals in the drift term. And so, taking that as a launching point, you can also do continuous time and just, instead of summing across a bunch of intervals, just take the delta t that's elapsed, and that's the variance that you've accrued. And sorry, there's a drift term here that I should have multiplied by delta t as well. So that's the easy step from kind of a bunch of integer, interval times to continuous time. You have to kind of, they introduce this new diffusion rate or diffusion coefficient parameter. The next step to add this, complexity is to think about this preference function. So the simplest way is replacing a constant drift term with this habitat preference function. And so the change over time in the location of an animal at a given time is the derivative of the preference function. And so, anytime people have a PDE like this, an ODE like this, it might seem intimidating, but the easy thing to do is just to do what's called a forwards Euler approximation. So you just take that gradient and assume it's constant over time and just multiply it by a time step. And so you get this sort of forwards solution to the over per movement over a delta t. Next you can do this in 2 dimensions, and the math is very similar, except you just have to replace the derivative with this gradient operator, the upside down triangle. So, for people that have not taken multivariate calculus, maybe that the new notation might be unfamiliar, but the concept is exactly the same. You just take a derivative of this preference function, in 2 coordinates instead of 1, and that gives you the direction you want to move. And if you're doing the forwards Euler, you just do one time worth of movement in that direction, and that gives you the drift, which gives you the update in expected location. Yeah, so that's sort of the simple, kind of a series of simple steps that get you through treating this sort of diffusion Texas model and how to simulate an individual walking across a landscape. And so again, in the textbook, we sort of simulate this case of a central place forager that tends to move away from its center location for a while and then comes back to it at time t equals 1,000. And so doing this, you can simulate a bunch of tracks where they go out on a 100 time foraging interval. You can fit that with just if you've got good data, you can fit that with diffusion only as a state space model, or you can do it with a drift term as a spline. The point being that you can kind of generically deal with that advection piece with splines if you don't have any better idea about how to do it. And so that's sort of the continuous space version. That's useful if you've got animals that have GPS tags, where we or Argos tags, or where we're continually getting good measurements of their location. We don't often get that. We get pop-up tags or archival tags instead for animals that stay below the water. And so instead, a different way to do this is by gridding, space and using this high resolution, discrete space, and you could call it a finite volume model, and you can solve it as a hidden Markov model. So again, kind of going back to concepts to try to bring people along with the intuition. If you've got you've got some kind of simple calculus here where the change in abundance over time is just this kind of linear rate of increase. You solve for it over time by taking e to the growth rate times the time elapsed, and that's how we compute compound interest in a bank account. It's how we do intrinsic growth rate. Presumably everybody is sort of familiar with the scalar exponential function. And so the exact same thing applies if you've got a vector of abundance across space. So you've discretized space into a bunch of polygons, and you're tracking abundance in each polygon. So you're doing this kind of finite volume model. And you've got this matrix A, which is the instantaneous movement rate matrix. And by convention, people often, take the vector of abundance and pre-multiply it by A. The general solution of this is, again, just using what's called the matrix exponential. So you take this instantaneous movement rate and you do this matrix exponential function that's in there, in RTMB or TMB or base R, and it'll give you the solution for a given delta t. So yeah, that's sort of the simple story there. If you've got this continuous time Markov chain with this instantaneous movement rate A, you can always just do the matrix exponential in R, RTMB. Then there's sort of a series of different approximations for how you can do this in different cases. So one of them is what's called the forwards Euler again. So if you subdivide your time into a bunch of subintervals, you've got this formula where you basically take a lower rate and do that lower rate movement over and over again. That'll maintain sparsity in some cases. The backwards Euler is basically doing the same thing, starting from your endpoint, and it has this sort of matrix inverse, and so it plays well with GMRF models. So if you've got some spatially correlated process error and you have a movement process, you can do this backwards Euler and you will still get a GMRF out at the end of it. And that's sort of the trick that underlies a bunch of spatial statistics. And then finally, there's this trick uniformization. So if you think about the definition of the exponential function, it's this power series, and you can just kind of compute terms of this power series until you get enough precision. So that's sort of the trick that is written into TMB and RTMB. You define the tolerance for errors you have, and it'll just automatically decide how many terms to include in this power series version of the matrix exponential. So, so that's sort of the, how do you do the math on this this discrete space version of that diffusion taxis process? So here's sort of the worked example from the paper I did with Steve Barbeau and many authors a couple of years ago. You might have on the top left in panel A, you might have a preference function that's increased preference as you go from 0 to 6, based on depth increasing. And then you've got a bottom temperature that's kind of lowest in the middle. And given some preference parameters, you calculate a preference function that's that black line in A. And that, just taking first differences of that preference function, you get this taxis matrix in C. The diffusion matrix is always sort of positive for adjacent spots and negative for your starting location. You sum together the diffusion matrix and the taxis matrix and exponentiate that, and that gives you movement. So if you start out at different locations shown in D, E, or F by these gray bars, so all of the mass starting at a single spot, you can calculate what happens after 1 time step or 3 time steps or 10. As t goes to infinity, it goes to stationary distribution, and if your domain is fully connected, then all of the animals will end up at the same stationary distribution. That's shown in the black line for every D, E, and F. And you can actually calculate that stationary distribution from the dominant eigenvector of the rate matrices. So that's how you get your stationary distribution efficiently. Just to kind of try to prove to people that you can do the math and have it be scale invariant. Here I'm showing sort of a hypothetical example with log elevation in Madagascar, made up log preference for a hypothetical animal that prefers sort of intermediate elevation, calculating the log stationary distribution, and then 9 times after hypothetical release. So the left-hand side is a high resolution model where it starts at this tiny dot in the kind of eastern coast of Madagascar, and it sort of moves outward over time to eventually get closer to the log stationary distribution by time 9. Instead, if you do it at a much coarser resolution on the right-hand side, you can see how the log elevation is coarser, the stationary distribution is coarser. But if you kind of squint, by time 9, you can maybe believe with me that it gets to sort of a coarser version of the same stationary distribution, the same density function, and also that the log stationary distribution is similar between the two of them. Okay, so that's sort of how you can do this discrete space diffusion taxis. We'll talk about movement later, but oftentimes we have these archival tags. So we've got an electronic tag that recorded depth and temperature and magnetic fields and light levels and so forth with a clock on board, and it pops off, eventually communicates by satellite. You download it, and using something about like the daily depth it got to and the time relative to its internal clock that the sun rose and set, you can get these components of a likelihood, that you then multiply together, and you call that the data. I mean, the tradition is to call that a data likelihood. So, just using the data on the archival tag in a given day, here's an animal that was released in the Aleutian Islands, and then the data likelihood for different days during this 90-day deployment. You can fit that model at high resolution. I mean, that picture, I think, had 40,000 cells in it, and you can give it bathymetry information, fit the movement process with this preference function and get confidence intervals, back out the preference function, back out the stationary distribution as the dominant eigenvector of that. And so it gives you this high resolution sort of Eulerian.
Just a reminder, there's only a couple of minutes left.
Okay. So finally, the third one is sort of doing this with densities instead of a single animal. And so again, the math of this looks similar. You've got this density vector across locations, and you move it around with this M matrix, which is e to the array matrix. You can have those sort of survival deviations like Matt talked about, and those presumably you'd want to be spatially correlated. And yeah, so you can do this in TMB. I'll probably skip the numerical stability part, but there's different parameterizations that make it more or less numerically stable. And so yeah, we did this with kind of a joint model of tags and CPUE and survey data and a seasonal model for Eastern Bering Sea, Pacific cod, and found that basically, as Matt said, if you don't have tagging data, it ends up, in my experience, very hard to separate out sort of the rate of movement just from survey data. And so in cases we've looked at without tagging data, we've sort of used the biology to kind of make some assumptions about the maximum daily displacement, and that's enough to kind of firm up the model. Yeah, so again, coming back to this sort of main point, one of the themes from spatial statistics is that it's much easier to do things at high resolution than back out coarsened, fit it to coarser data. That's called a change in support model. And similarly, if you've got a theory that gives you movement in continuous space and continuous time, you can fit it to continuous tags, in continuous space. You can fit it to high resolution space with archival tags, or you can coarsen it. But it's much harder to go the other direction. And so I think if people are, willing to spend the time thinking through this sort of diffusion taxis movement piece, they can get, dynamics that are applicable at a, high resolution or coarse resolution. So I'll put up a list of people to thank, and thanks again.
Okay, thanks, Jim. You used up all your time, so I think we'll move directly on to Ina, which I think she's going to cover some aspects of moving between small and large scale models as well. So I think that'll be interesting to have that first before the discussions. So Ina, can you share your screen? Oh, you have, yeah. We can't hear you.
I'm now on. Okay.
Yeah,
can you hear me, see me?
Yeah.
Good. I'm just going back to my presentation. So do you see the large screen?
Not yet, no.
Not yet. Now?
Still not. Oh, we can now, yes.
Okay, good. Okay. So thanks for inviting. Happy to present how we do the quantitative modeling of the spatial temporal dynamics of 4 major tuna stocks, estimated distributions of which you see on this screen, on this title page. And I would like to acknowledge our team, the dedicated team that works hard on the CPDIM modeling, quantitative modeling. So to introduce to CPDIM, I thought it would be good to highlight and to show you the the history of the CPDIM development as from the classical advection diffusion reaction equation to the quantitative model of population dynamics. On this timeline, you see that the model has been developed for nearly 30 years, starting from the So just as a different phases of development started from the simulation advection diffusion reaction model, then adding the fisheries data to be able to estimate model parameters, then adding fisheries tagging data in addition to the fisheries data, and now we are having the full likelihood, including the catch links, tags, and early life data in CPDIM, and posing the question whether CPDIM can be used as the next stock assessment model. So I structured today's presentation around this question. So let's dive into the basics, well, not basics, but into the math of the CPDIM, and thanks to James for presenting the basics of the advection diffusion reaction models. So underlying equation of CPDIM is advection diffusion reaction. I'm just trying to, we'll take the pointer to show. Okay, this advection diffusion reaction equation describes three dynamic processes: reproduction, mortality, and movement. And it has age structure. So you see the terms of this equation, they describe the change of the density. Well, first of all, the model describes the density of fish population in 4 dimensions, in age, time, and two-dimensional space. And this density, so the term describes the variation of density in time, in age. Then the advection term that includes both passive and active velocity. So the active, which is, as James mentioned, driven by the gradient field of external biophysical force. Then the diffusion term, where here diffusion is the space varying, it's not constant. And mortality, including both natural and fission. This equation is complemented by the initial conditions for density and 2 boundary conditions. So the reproduction term S is represented by the boundary condition at age 0. And as the spatial boundary conditions, we can use We can use Newman as shown here, which is closed boundaries, where we can imply the closed boundaries when we say that tunas or other species cannot go across the boundary because they are physically, like, I don't know, the land, for example. Or if not, then we can use the Dirichlet boundary conditions. And this is the most classical advection diffusion reaction equation, even with age structure is classical. I listed here the recommended literature that describes in depth how these equations are derived from the individual movement and so forth. So please read those if you're interested. What kind of movements this equation describes, because it's a key process. I think it's important to highlight that, as I said already, there is a passive and active movement. So the passive includes the transport by the ocean currents. Besides, this transport is not only applicable to larvae, because even when tuna are larger, they're still impacted by currents. It's just their velocity is changing through impact of currents. Then the directional movement also includes the movement towards habitat. And here the good example is if we easier understand the movement of individuals than the density. So the example shows how the individuals would be distributed in the finite time if we let them move in the heterogeneous field, like a habitat here, the habitat which has most favorable condition in this location. So in a finite time, we will see that the majority of individuals, maybe they will all be concentrated in the good habitat. And this phenomenon is modeled in the advection diffusion reaction framework as the movement towards the gradient of the habitat. And this is how we do it in CPDIM. And about non-directional movement, so important to highlight that we do not model the movement of the scarce population density populations like whales. which have very low densities, and they are better modeled, and their model is better approximated by the represented by the individual based models, and they cannot be approximated by that advection diffusion reaction. Only the large number of individuals can be described and successfully approximated by the diffusion term of this equation. Okay. Coming back to the model itself, important to understand that CPDIM is a numerical solution of underlying advection diffusion reaction equation, and this solution is obtained with help of the advection of the alternate direction implicit numerical solver, which was developed back in 1994 by John Seibert and Dave Fournier, and CPDIM benefited from this work. So we still use this solver. Now what? We have a nice simulation tool, but we want to estimate those dynamic rates, velocities of advection, diffusion rate, and mortalities, and reproduction rate. So we informed those rates. So we informed our modeled processes by the data. And as it was already said, in the spatial temporal data, we need the model, we need the georeference data. Two types generally we use in CPDIM to inform parameters is industrial fission data and scientific survey data. So industrial fission including the catches and effort and catch data. Historical, and ideally they should be complete. So the georeference data should match the landings, the total landings, and the length frequency data, which are also georeferenced, but at the lower spatial resolution, like we can have 5x5, 5x10, and so forth regions covered by the length frequency data, but we have a good time series of those. And the scientific survey data includes conventional tagging data that we use to inform our movement parameters, and two types of early life data. First is the X survival data, which is, for example, for skipjack, we are using the empirically derived relationship between temperature and the survival of eggs hatching and hatching times. And we have the densities of the early larvae, very small larvae of several millimeters that were observed in the surveys back in 50s to 80s by Japanese scientists. Okay, we know that fission data are good for informing reproduction mortality rates and the spatial extent, but very poor or even do not inform the spatial distributions and movements in spatial dynamics. And this kind of data are essential to estimate model parameters. So how do we estimate? Of course, in order to, before going into the estimation, we need the observation models. I think you are all familiar with the observation models for the effort, for the catch and length frequency. Maybe something new that can be used in the mechanistic process-based models like CPDIM is more physical approach is when we predict catch given the density available, and it's called density biomass removal or catch removal method. Although it only observes provides information to the model when there is not enough density locally. And for tagging data and early life data is not that easy. We need to solve additional equations to observe tagging or early larvae. I will not go into details because I feel that I will not have enough time. The parameter estimation is done just as well as in the stock assessment modeling with the maximum likelihood estimation approach. What are we estimating? We are estimating a limited number of parameters. So here is the list. In the parentheses, you see how many parameters. Why they are so few is because the temporal dynamics, actually the parameters are representing the responses or the links to the environment. Responses to the environment, the link to the environment, how it impacts different processes. For example, feeding habitat, we have several parameters which describe the thermal preferences of tuna, the food availability and thermal preferences, age dependence, the mortality rates include the predation, senescence, and starvation, and so forth. The parameter estimation workflow itself is more complicated than just more complex than just estimating parameters given this formula, because we want to know whether the parameters can be observed from our data. Because we need to have, since the model is has higher dimension, we need to observe the processes across all dimensions. So across age, for example, we need the observations for larvae, and if we don't have it, then we have a gap between the age 0 and the age when the population is recruited to the fished stock when we have the data. So we might exclude some parameters and fix them. And for this, we need to do a bunch of preliminary analysis, which include so useful tools such as global sensitivity analysis, variational analysis, or identical twin experiments, correlation analysis, to be able to exclude those parameters which are correlated. Okay. Yes, the point here is that the parameters that are estimated in the spatial temporal mechanistic model are normally time and space invariant, and this is tested applying statistical scores to the independent data. Once we get MLE, we can verify whether this solution is still optimal, is still suitable to fit the data, observed data outside of the time window used for the estimation. And for this, we have a bunch of metrics for the catch lengths, particularly for the tagging data, including the explained variance, variance ratio, normalized mean square error, or root mean square error normalized by standard deviation, the Kling-Gupta efficiency metric, the pattern correlation, as an example. And of course, we have some established some thresholds to say that this is valid or not, which is useful. So coming to the main question, I remember, Mark, you asked me when we were at this workshop in DTU, how is stock estimated in CPDIM? Well, in fact, it means how do we know that in our estimated distributions of the tuna stocks, we have, for example, 1 metric ton per square kilometer of skipjack in the fishing ground, but only 30 kilograms per square kilometer of albacore. So we actually, if we don't do any assumptions about the stock recruitment relationship, about reproduction rate, then we cannot estimate optimal stock. What we can estimate the minimal stock size that sustains observed fission pressure, given its spatial temporal age distribution. For example, here I added 2 plots to illustrate this approach. We are fitting these densities of skipjack to the Persane catches. And so we can see that the density of the total stock of about 4 million or 4,000 tons is enough to sustain the Persane catches object associated. So enough means that we fall below the threshold of 1% observations with relative error greater than 10%. When I say percent observations, I mean the locally at 1 degree scale. And here the black curve corresponds to the MLE. So during the the max parameter estimation window, we see that 4 million tons is enough to sustain persane objects. However, when we go to the validation, which is 2013, 2022, this stock is too small. So we need to increase it to 6 million. But we do it for all fisheries combined. And we see that free school persane tells us that we need at least 7 million ton for skipjack to sustain the catches combined. Okay. And yet, when we compare our estimations to the multi-fan-cell estimates, we are systematically above. We have our abundance was never observed to be lower than those estimated by multi-fan-cell. On the left, there is a comparison between multi-fan-cell and CPDIM, so multi-fan-cell red, CPDIM black, for recruitment, all regions, and total biomass, all regions. The good news is that we are converging. So if you look at the assessment comparison of the same solution with the assessment 2025, we are getting very close, although above in terms of the total stock estimation in WCPO, but very closer, and we have the trends converging as well. And interesting bit that I wanted to share here is that the change that occurred to multi-fan-cell assessment is that now they took into account the effort creep of the Japanese pollen line fishery, which was demonstrated in earlier work that there was a fourfold change in catchability estimated. And actually in CPDIM, we don't have this problem because in the mechanistic model with the estimated link to the environment, we have this change naturally. Like in 95-2012 MLE window, so parameter estimation window, we have a constant. The model was able to fit to the Japanese pollen line data with the constant catchability, no change. However, when we re-estimate the catchabilities for the period 70 to 90, we see that it was changing. In order to fit to the densities without changing the biological parameters, we need to increase the catchabilities. So the question that was asked also is how CPDIM can be, and actually in some cases is already used externally for stock assessment. First example is the tag mixing simulation studies that were done with the individual-based model ICAM-1 by Joe, where he took the parameterization, optimal parameterization obtained for CPDIM. So simulated CPDIM solution with the individual-based model. So having this baseline solution for the entire population and then releasing the tags. So for the subpopulation, he was able to figure out how many months, quarters needed for the good mixing. And for this, he was using tracing the recapture probabilities and using the dissimilarity tests to see when the distribution of the entire population, or the recapture probability, sorry, of the entire population and just the subpopulation, the tagged subpopulation were matching. And the conclusions were that the mixing varies greatly between regions, and using CPDIM, specific mixing periods can be assigned given the release group. Another application or another case where CPDIM can be used externally for stock assessment is the learn from CPDIM on the environmental variability of recruitment. So, for example, provide recruitment indices or regional abundance indices from CPDIM to the stock assessment model. As an example here, in CPDIM, we observed that skipjack recruitment is highly driven by the primary productivity. that is confirmed by the increase of observed catches in this period as well. And third example is estimation of the movement for the use of estimated movement to derive the block transfer rates. With CPDM, we can compute the flow rates from one region to another. For example, this one, the beta, shows the total regional biomass moving from region 1 to region 2 during the given period from T to T plus delta T. And we can construct this matrix of the flow rates for a given multifacial regional structure, where the row in this matrix represents the entire biomass, the biomass in the region before the move, and the column is after the move. So we can compute then the probabilities of the movement from one region to another as the beta divided by the sum across the row of the matrix. Yeah, I think I will, if there will be questions, I will be happy to answer and illustrate this nice Hofmöller diagrams showing the movement. So to conclude, I formulated some pros and cons of, I suggest considering some pros and cons of CPDM usage suitability for the stock assessment. So as they actually can be considered as a line. So the pro of such model is that we have low number of parameters. We know in stock assessment, there are, well, as long as I know in multifacial, it can be several thousands. And however, in our case, the ocean physics, the ocean biochemistry, also combined ocean forcing can be considered as the fixed parameters. And so our assessment is sensitive. It depends on the quality of ocean forcings. So it's at one side it's a pro, and another side it's a con. Population dynamics parameters, fully validated reference model are space and time invariant. However, the overall development of this reference model is costly due to high dimension. Reference model is not sensitive to adding new fisheries data. However, it is sensitive to the changing forcing model because every ocean model has biases. Quite important. Another pro is that this model predicts unobserved biomass due to process, and this is very good because the biomass that was not observed through catches at time t, it will be observed at time t plus 1 because it was moved from another unobserved area thanks to the process. However, the same model has the diffusion-induced cryptic biomass, and this is a bad thing because it is linked to the high advection rates and the low grid resolution. And 2 degree, 1 degree is the low resolution. Another pro is a list of advantages is that we are free of typical problems, some problems that are quite nasty, like this example with the effort creep, and here it's emergent due to the link to environmental variability. The stock recruitment parameter is usually well estimated locally, especially when we use larval data. It's very well observed. No CPU standardization usually needed except for the vessel effect, but we don't have those information spatially. So we are not using standardized CPU. And we also, since the model reproduces the spatial heterogeneity explicitly, we can capture the bi or multimodality of the length frequency due to regional effects. And of course, the con corresponding to this is that we need detailed georeference data across all model dimensions. Thank you.
Okay, thanks, Nina. So I'll open it up for discussion now. So any questions to any of the presenters or any comments in general on spatial structured models? Is there anyone that wants to start the discussions? don't see any hands up. So I'll start with a question on going from fine scale to coarse scale models. Do you have to take the distribution of abundance within the large scale area into consideration when you are doing these analyses, so that you know, if the fish are all aggregated in one part of the large area, is that going to affect how you do the modeling?
Well, usually for parameter estimation, we tend to select the time period large enough that is the most covered by the data. For example, for skipjack, I select the window 91 to 2012 because we already have the Procyon well developed. The estimation starts from 95 because we still have the pollen line fisheries and we have the large spatial extent. Of course, if, as you said, we will only observe a part of the ocean and nothing in the rest, it is useless. This data, we need good spatial coverage, sample spatial. Did I answer your question?
Yeah, I think so. What about Jim, with the methods you're using? Does that affect those as well, or is your method sort of general that it's applicable no matter what resolution you use?
So, yeah, my claim is that, you know, and we heard this in the other talks too, you know, that, you know, you can use that advection diffusion or reaction advection diffusion regardless of scale, and you can parameterize it to be independent of scale. I thought what maybe you're asking about is if you fit a model at high resolution and then you want to take the output and plug it into a coarser resolution model, like maybe you fit tags at high resolution in one component of the likelihood, and then you want to back out coarser movement fractions that you apply to pop dye in a different likelihood component. Is that sort of what you were talking about? That sort of
That's one thing too, but also what about the initial conditions, right? I mean, in a large model, you might have 1 billion fish in that large area, but if you went to a finer scale, you might have 1 billion fish in one corner of that large area, right? And with I mean, is that important when you're comparing these two different scales? And if you're fitting to data from different scales, like say you had tagging data where you know exactly where they were released, where you have other data that's sort of on a broader scale. I don't know.
So for instance, I think, you know, I think Matt started to talk about this. You know, my claim is that if you have one likelihood component that is high resolution gridded archival tags, and maybe you have a different likelihood component that's continuous space, you know, this like Lagrangian state space reconstruction of animal movement with GPS tags. You know, if you're going to coarsen that to, you know, a few box model, big box model as a third likelihood component, you need to know the kind of distribution within those coarse scale to get the kind of weighting of the fine scale fractions right. So Matt had a slide where he had an equation from that paper I did to Steve, and that's essentially what that sequence of, you know, it's like 3 equations in the paper was doing, is just doing that kind of biomass weighting for fine scale movement within a coarser box to get the coarse scale movement.
Anyone have any questions? Yeah. Carolina?
Yeah. I have some questions for Nina. So, hello.
Yeah. Thanks, Carolina. Hello.
When you compare the CAPAM with the stock assessment results in 2024 and 2025, in 2024 they were much different. In 2025 they were similar, and you mentioned it because of the effort creep, right?
Sorry, Carolina, can you please speak louder, or I didn't quite hear everything.
Yeah, so the only, when you compare the assessment model results with the CPODIM trajectories in 2024, they were very different. And in 2025, they were very similar. And you mentioned it was the only difference was that the effort creep was addressed, right?
Yes, exactly, in the stock assessment, but not in CPD. It was not included during the parameter estimation, the effort creep. During the parameter estimation, the catchability was constant for those fishery, but it could be estimated once we validated the model. So during the validation, what is the validation? We have estimated all the biological dynamical parameters. Fisheries parameters, they are time variant. So they are different during different times. So we want to re-estimate fisheries parameters by having estimated the biological parameters, but by keeping them the same. The biological parameters aren't variant. So we just re-estimate the fisheries, and we see that the catchability of those Japanese pollen line was increasing gradually. And this was actually explained by the changes in the fishing practices.
Okay, thanks. The other question I had is, like you mentioned, the CPODIM is sensitive to the ocean models.
Yes.
So how sensitive? Like
it can completely drastically change the model solution if you change the model without re-estimating parameters. And like if we are using the Earth climate models, which have huge biases in terms of the temperature, like a very simple example, we estimated the preference of skipjack tuna being 28 degrees at the larval stage, okay? But in the Earth system model, we have systematic bias towards higher temperatures. So if we will be taking our 28 degree, the spawning will happen in different area, simply.
So I was wondering if in that layer it could be like it could be like combine like a spatial temporal model, include like the random effects in that layer to be able to accommodate for bias
for the biases, but not for
different
systematic biases.
Not for systematic bias, yes. So to be able to use both, kind of combine both approaches into one. Well, yeah, thanks.
I think it's Mark Bravington. That's the question.
Okay, can you hear that?
Yeah, we can hear you. Yeah.
You can probably see me as well now. Yeah, can I have two questions?
Sure.
Is that a bit rude? Thanks. Okay, the first one, kind of for Jim. So you started off by talking about physicists model particles, and that's what physicists do. Does it bother you that fish are not particles? I say that with an eye on things like the bluefin tunas, which are also tunas, I would like to point out, and they have, quite specific migration patterns, often to come back to spawning grounds. I don't think we really know, but I wouldn't be at all surprised if they develop individual preferences for where they go to feed when they're not busy spawning. And so the models you're talking about have particles which essentially are memoryless. I guess the implicit assumption behind everything that's being talked about here is that the memory of fish doesn't matter, and they don't have sort of persistent individual characters that are going to matter in the kind of assessments you're talking about. I say this, of course, with half an eye on close kin, but I'm not sure that bluefin behave as the particles you're talking about. And so I just wonder what we think about that for other tunas, albacores and the tropicals. I'll stop there while I try and think of the second question.
Yeah, no, thank you. I guess my talk abstracted away from a lot of details, and I'm just trying to sort of encourage people to take the plunge to think about PDEs. I don't know that everybody does that in the field, and so I'm sorry that I kind of skip a lot of details, but I do think that for instance, like seasonal movement, you can get at with sort of like cyclic spline. I think you can get at with cyclic splines on the preference function, where sort of advection, if you drop out the diffusion term, an animal will tend to get back to the same spot. And then, if you've got sort of population mixing, like Matt talked about, you might have to model a mixture of different source populations. And so I don't mean to minimize a bunch of different practical issues that I think Matt did a better job of summarizing, like, if you've got different behavior for different natal source, or, you've got age varying or sex varying, age and length varying. But I do think that those types of cases can be handled by sort of plausible extensions of advection diffusion models.
Oh, okay. Thanks. Can I do my second one now? I think I'll just try and do it. It's sort of for Mark, but for everybody. Jim as well. Is it so early on when Arnie was summarizing, I think what Mark's proposal was, the word simple appeared in it. That simple has been not quite so evident in the talks here, because they're quite complicated phenomena. And I wonder if could somebody summarize what you see as the main purpose of the spatial element when it comes to tuna assessment? Like, I think there's in here an implicit assumption, which might be fine, that you're looking at single, not exactly panmictic, but populations where you're trying to look after the numbers in the whole stock. I think that's an implicit assumption of how it's going to be used. So is the spatial model there to kind of make sure that we don't have inconsistency between removals and taking, more fish out than there are in the sea locally? Is it in there to kind of fill in holes in the CPUE coverage, or what's its sort of principal role that you see in the kind of answer that you ultimately need to come up with? Over.
I'll have a stab at answering that question. We do see spatial structure in the tuna populations. Some of the tunas have specific migration patterns, like bluefin. The North Pacific bluefin does the same thing, or it goes from Japan to the US and back, things like that. So obviously that's kind of important to think about. But in the tropical tunas, which we're kind of more worried about, because that's our the biggest stocks that we're dealing with, we see spatial differences and we see spatial movement, but we don't see the same migration. Right, we generally see more of an advection diffusion type process rather than annual migrations or anything. We also see differences in biological characteristics, like the length, maximum lengths that we see in different areas. And that might be due to movement, but it also might just be due to different growth rates in different areas and limited movement, right? We also see some possible stock structure patterns where some areas we see, if we look at the recruitment rates, they're correlated with El Nino, and in other areas they're not, right? So there might be environmental things there. So one way to do the modeling is saying that, well, the movement's fairly limited, so we just can break the population up into whatever discrete units we want, and we're approximately correct. So by doing that, we might break it up by management areas, right? So then we don't have to have problems with trying to manage internationally and things like that. Okay. So there's a whole lot of spatial issues there. One of the big spatial issues that we think is important is the tagging data. And the reason for that is to tag fish conventionally, they've got to live. So that means you've got to find a way of catching them and letting them go so that they survive. It's a very large ocean and fish are everywhere. So again, it's hard to get out there and tag everywhere as well, right? Because it's not as practical. And so tag mixing is one of the big problems. And so we'll talk about that in the afternoon, where if you don't take the tag mixing into consideration, you might get large biases. And so looking at a fine scale spatial temporal modeling, you can deal with the tagging and you don't lose all the information, right? So some of the ways we did it in the large block ones is you say, we estimate an F for the first 6 months or something, because the fishing is located more concentrated where the fish are tagged, right? But then you're throwing away 6 months of the data. So, you know, the skipjack's already dead, so you have no data. So there's a whole lot of aspects of spatial modeling that are important to consider. And things like local depletion might occur, so that might be important for management as well.
Maybe to throw in a couple of comments on and on this initial pitch that Mark did in July, he in an email format, what he was contrasting was a few choices to be made. So in terms of spatial structure, it can either be fine scale, sort of continuous spatial temporal kind of approach, or just these block regions that we've been using for decades. And in that email, he said, yeah, let's just go with the block regions. And then he went on with, as for tags, we can either try to do something very detailed and sort of statistically sensible, like multi-FNCL, or just forget about tags and just do all the tag analysis externally. And he said, let's just go for that. So that's what I meant by simple, that it's just big block regions, no tags in the model whatsoever. However, all of this is pragmatic if we deal with the tags in a fine scale spatial temporal model as they did with the skipjack last year. From a statistical standpoint, it would be better to have everything within the model as opposed to sort of breaking it into 2 steps, analyzing the tags externally and then incorporating them as abundance indices. I suppose it's just the sparseness and caveats of the tagging data and all the other data we have that brings us down to pretty blunt basic approaches, at least for version 1. But yeah, and maybe just one more comment that still we are enormously interested in the newest developments on spatial structure and movement, just not to turn a blind eye on where research is going and considering that as an add-on for later versions.
And to put a little bit of context with that, my recommendation was based on if I had to make a recommendation at that point in time about any knowledge. And it was basically thinking through the problems, like what's going to cause us the least problems in developing a model. And I think that's part of it. And that's why we've got this workshop, is to see if that approach is the best approach or another approach is better. Like, for example, you know, going for the Cepodime approach, or maybe collaborating with fins and things like that. Jim, you had a comment on simple.
Yeah, I guess I wanted to clarify, because simple was in the title of my talk. Of course, like, claiming simple and complex is sort of a rhetorical point, but I guess my intent would be to have some kind of method that's easy to, that results in very short code, basically. So you can have sort of an easy snippet of code that you can plop in different places that works across different resolutions. And so, that vection diffusion piece in the matrix exponential is very compact code And I think it sort of fits with Arnie's sort of initial claim about, having sort of a small RTMB code base. I realize it's complex in other definitions of complex. Yeah.
I mean, one question related to this is like, can you take that particular approach and apply it to a 2-region model and a 100-region model the same? I guess that's a yes, is it, Jim?
I'm trying not to. I'm seeing if wanted to jump in, or Matt, but yeah, I mean, I haven't pushed it that far. I mean, in the cases I've looked at, I've compared like the 10,000 versus 100 cell. I don't see any fundamental reason you couldn't do it at even coarser resolutions. You'll end up getting weird artifact. The approximation error gets more. It gets greater as you do it with coarser boxes, but the math applies the same way.
I mean, Jim, we've done it in that paper we're working on, where essentially we have the same PDE and we're modeling movement as continuous, but then essentially it's just large box strata, and we still have the same sort of fundamental equations there. So I think it is possible to approximate those dynamics, but I'd be intrigued to see how the total sort of movement rates estimated from a fine scale versus spatial, or fine scale versus coarse scale model sort of comes out. But yeah.
Like, is CPADIM able to change the resolution? Like, do you have anything to say about what happens when you
Yes.
change the resolution?
Yeah, I wanted to intervene in this and add a comment. Actually, yes, we do play with the resolutions. We do estimate on the 2-degree and 1-degree scale, and meaning that we have uniform grid with the size of one cell, 1 degree or 2-degree. And we know that coarse resolution yields in the higher stock because of those numerical diffusion involved, or even not the diffusion, but the size of the phenomena described. Actually, the size of the phenomena, the model that is coarse resolution, it can only describe the coarse distributions, if you want. So you will always have the rest of the distribution, which is completely cryptic and observed. So you need this high peaks in the distribution, the high resolution phenomenon to be observed. Sometimes it depends on what species you are considering. So when we go to the coarse resolution to the finer resolution, we usually have the reduction of the total stock size with the same fit to the data.
Okay, so it's time for the break. So we're going to break for 20 minutes and come back at 1:10. And we're going to start up on the tagging data topic, which is very related to this one, so we can continue some of the discussions from this earlier period then as well. I think that if we want to. We're going to start up soon. Moving on to the tagging section. Dan, are you there?
I am. Can you hear me?
Yeah, we can hear you clearly. Yeah. You can try and share your screen. Yep, I can see that.
All good?
Yep, looks good. Full screen too.
All right.
So yeah, feel free to start.
Okay. I'll give it a go. So yeah, I'm Dan Gaethel. I'm going to be talking about integrating tagging data. I will say I've never worked on tuna models, so I don't know how Mark keeps convincing me to do this presentation on tuna tagging, but it did give me a chance to kind of dust off some family photos, albeit of bluefin tuna in the Atlantic. So maybe not the right species, but it's always good to have some pictures. And yeah, I just want to acknowledge Matt Vinson, Matt Chang, and Aaron Berger, my co-authors who helped with the presentation. And yeah. So hopefully I can kind of skip some of these slides or go through them real quick since we talked about a lot of this stuff, and I think it's kind of basic to most people that work on Tuna. But we do have kind of, 4 general types of tagging that we could be integrating into a stock assessment when we start talking about spatial stock assessments. They each have their pros and cons. So we've done a lot of talking this morning about conventional tagging, which are useful because they're low cost, but they're also kind of low resolution, as we talked about. The next scale up, you kind of have electronic tags where you have kind of these low sample sizes because they're really expensive to buy and then to deploy. But that gives you a much more high resolution kind of tag tracks that can really start informing some things like preference functions and other types of movement like Jim talked about. Kind of the in-between there is these natural tags, either parasites or otolith microchemistry, which are nice because you don't really need to deploy them. They're just naturally occurring. So you kind of get past that deployment part. The issue there is that you need to know a lot about the life cycle of the species that you're studying. And then finally, genetic tagging, close-kin marker capture, which are really nice because it can give you a direct estimate of biomass, but then you have a lot of cost associated with extensive sampling effort. Just FYI, for the remainder of the talk, I'm kind of going to just not talk about the CKMR genetic tagging stuff. There's a whole session kind of devoted to that later in the week. So I'm going to focus mostly on the conventional electronic tags for this presentation. And again, we kind of talked about this already, what does tagging inform? Basically, you get information on migration, on population structure. When you start doing tagging models, you can get information on growth, on mortality, things like that. And I think this is an important point. There's kind of 2 ways to handle the tagging data, which was just kind of discussed in that previous discussion, which is you can either externally kind of analyze the tagging data in a tagging model, get your estimates of, say, movement and mortality, and then put that into a spatial stock assessment if you wanted to, model movement and things. But you do have this kind of problem where you have 2 different modeling frameworks. So then you have sort of a loss of information, but more importantly, kind of this inconsistency in your assumptions, and then some difficulty trying to figure out, generally what the uncertainty is as you move between 2 frameworks. So I'd say like kind of the gold standard in terms of doing kind of these spatial assessments is doing a tag integrated model. At the very basic basis of a tag integrated model is you just have this tagging submodel where you're modeling the tagged individuals. You put that into the overall assessment, you follow those tagged individuals through time, and by fitting the tag recaptures, you can simultaneously jointly estimate parameters like movement, mortality, potentially biomass, depending on how you formulate it, and same with maybe growth and maybe tag reporting rate. So just, really briefly, what is a tag integrated model? Well, basically, if you have a spatially stratified model like Matt was talking about, you're modeling multiple independent units, or semi-independent in this case. So in this case, we have 3 kind of populations. This is for yellowtail flounder for my dissertation. But basically, the tag integrated part is at some point during the time period, you're going to have a tag release event. So then you mark individuals wherever they are at that time. And then, at the end of the year or beginning of the year or continuously throughout the year, depending on how you set up your framework, both the unmarked and the marked fish move. And then when you have those marked fish, you can keep track of where they are and where they move to, and that helps you obviously estimate movement. So underlying the basic framework's relatively simple. And then previous CAPAMs, we've talked about spatial models and tagging data pretty in depth. 2018 one, which was on spatial stock assessments, kind of noted in the summary paper that tag integrated models generally perform best, but there are a lot of challenges to using tagging data into setting up spatial assessments. And I think kind of the summary to me is kind of that generally, I think we need tagging data to estimate movement and do a spatial stock assessment if we're being realistic, but that we have a lot of concerns about using tagging data. Some of them, I think, maybe we overthink at times. Just when you compare tagging data to general fishery dependent data, all our data kind of has problems, and it's all relative, how informative they can be. So just to summarize, specifically for tuna assessments, what the primary kind of issues are for tagging data. I think the overarching issue is whether the data that we're able to collect is representative of the entire population. Again, I think Mark mentioned this, like you have these huge ocean basins, you have these fish that are widely distributed, they're highly mobile, they cover great distances, and it's really hard to go out there on a boat and try to sample across this huge region. You have to catch fish, you have to put them back in the water alive. And if you're using conventional tags, at some point you have to get a recapture before they die. So there's a lot that goes into it, and it's really hard to kind of get that data and make sure it's representative of the whole population, or at least a population in a certain area where you can make some inference. And then the second issue that comes out of that, as was also mentioned, is tag mixing. Once you start, aggregating up to these big spatially stratified assessment units, you have to make these assumptions about how well mixed tagged fish are with the untagged population, how to make inference from that. It becomes really difficult at these big scales. And then, moving kind of down the list, you have, trying to figure out what your reporting rate is. You're relying, for the most part, on fishery recaptures. So you have multiple fleets, multiple flags, and you have to kind of figure out, what that reporting rate is, because that can add some bias if you're misestimating reporting. And of course, there are a bunch of other kind of we term nuisance parameters, especially with conventional tagging data. So things like release mortality, tag loss, those are all very important when you start trying to infer what the mortality is and the movement. And if you don't know what that is, or you incorrectly, put a number on that, then you start getting a lot of bias. And then, obviously the goal for a lot of these tagging studies is to estimate movement. And so if you have non-representative samples, or it's difficult to scale up from the individual to the population, then you might start getting unrealistic estimates of movement, especially when you start aggregating to these big spatially stratified units. And then just generally, I think these are issues kind of for all data sources, especially in the spatial models, is data weighting. We have this kind of new, I won't say new, but kind of different data source that not everyone in the assessment world uses. So when you start thinking about weighting across all these data sources, how do you deal with that for tagging data? And if anyone that's worked with tagging data and trying to fit it in an assessment, the diagnostics for assessing the fit become really, I'd say, kind of tedious and hard to really visualize. You start looking at multiple spatial strata, multiple cohorts, things like that, and trying to look at, say, a residual pattern for tagging data becomes really difficult in comparison to, say, residuals from a single region survey or something like that. So I'll just try to kind of briefly go over some of the main issues. Like I said, representativeness, I think, is the main one. You either have few releases or few release events for the most part, depending on your tagging type and, when you're talking about tuna, like I said. So then you kind of have limited coverage, both spatial temporally, but even more, potentially more importantly, by life stage, especially if ontogenetic movement patterns are important, then you obviously need to cover all the life stages with your tag releases. And then obviously accounting for your heterogeneous fishing efforts important, because where and when you recapture fish, has important impacts on how you get your movement estimates. And so, mainly it's just whether or not your tagged fish represent the dynamics of the entire population, and whether you can get kind of unbiased inference of population level processes. I put solutions, but honestly, these are kind of, I don't know if there are solutions to any of these, but there are ways to try to deal with them. The representativeness issue, I think, is really just a monetary cost constraint kind of situation, where if we were able to go out and tag across an entire ocean basin, then we'd probably be not worrying as much about the tagging data, but obviously that's kind of impossible. So the more feasible thing is, kind of using multiple tag types and using the power of both types of tags simultaneously. You can kind of adjust your assessment units to maybe better match the tagging locations and better deal with tag mixing. And then, like we talked about, there's a lot of up and coming high resolution, both simulation estimation approaches that can help you kind of infer the fine scale dynamics and kind of move from the individual tag tracks to the population level. And Ben Gallardi has some interesting work with that with his SAT-TAG-SIM R package. And then moving kind of to these nuisance parameters, obviously the incomplete mixing is one of the bigger issues. The problem there is just the probability of capture differs, and it can induce bias in all your estimated parameters. The tag field issues, so your tag fails, you got mortality of fish, loss of tag, all those things can bias your estimates of mortality, specifically M, natural mortality. And then your non or misreporting, obviously that's problematic and it leads to underestimates of fishing mortality and or potentially movement. So this was also mentioned earlier with the conventional tags, when you have kind of this issue of mixing within these big boxes. One way to handle this is try to estimate your mixing period and then censor data within that mixing period until you think they're well mixed. But then you start losing data, and that can be problematic, you know, if you have a shorter lifespan fish, especially. You can also estimate different Fs and other parameters during the mixing period to account for incomplete mixing. But these are all kind of suboptimal, to be honest. With the tag field issues, obviously you can do simultaneous or post hoc analysis or studies like double tagging, holding studies to try to get at some of these parameter estimates. And then obviously misreporting, you can do things like high reward tags, seeding experiments, and try to estimate what the reporting rate is from a given flag. But you know, all of these things are pretty difficult to do in practice and get reliable estimates to use in your model. So I think, especially for the tag mixing, I think this is where the high resolution tagging models come into play. I haven't worked on any of these, but you know, reading the papers, they seem promising and they're really interesting to look at. Essentially, we have this hybrid spatial temporal model with your small grid cells that allows you to make assumptions of tag mixing on the order of days as opposed to months or years. And I think it really helps maximize data content because you don't have to kind of censor your data like you do for a big box region. And I think my impression is that it really helps you better identify kind of these preference functions and estimate movement at these fine scales as they link to, like Jim was talking about, advection, taxis, and diffusion. But I think we heard this morning, you know, there are high data and computational demands that might make it a little bit difficult to put them operational in a large tuna model. I'm not 100% sure that's, you know, that's Jim's ballpark. He can answer those questions. But I do think, and this was, I think, mentioned by Inna, is that you also need these really good spatially resolved environmental fields, and small changes in those can likely lead to kind of big changes in your movement estimates, which will change, you know, trickle down and change your biomass by region. So I think those are all considerations that you need to take into account. And again, we kind of already talked about this, but there are multiple ways that these could be used. You can do them to, say, estimate movement externally, scale that up and put that into like a spatially stratified assessment unit. There are examples. I think we're going to hear from Tobias in the next presentation about his efforts with all of this. But essentially estimating kind of fine scale biomass by integrating tagging data with catch effort and I think comps. So that's an interesting approach. And then obviously Jim talked about just integrating these high resolution tagging movement models within a spatially stratified assessment where you can then scale up to the movement from these spatially stratified units, which I think is useful in that you have a single kind of framework and you don't deal with the loss of information and difficulty estimating uncertainty, but then you might run into some constraints in terms of runtime and things like that. So one question I have about these high resolution models, since I haven't really worked on them at all, is whether we can really get away from this issue of the spatially temporally limited data. If our tag data is not really representative of the entire population, does that actually lead to a better estimate of movement just because we have a more complex high resolution model? Having said that, I do think they're really useful in a number of ways, and I think we should be using them more for some simulation experiments to test some different more aggregated assessments to see how well they perform. But obviously, we heard this morning there's a lot of different ways to use these models, so it's really interesting avenue. And I will quickly take a time to give a shout out to this yellowfin tuna simulation experiment that we did back in 2023. And really, why I wanted to talk about this is because we used a high resolution the spatial population model, SPM, as the basis of the simulation, and then applied different aggregated stock assessments to data from that spatial population model. And we found some interesting results. And so the two main papers from this are listed there at the top. Aaron's actually came out this morning, so if you're interested in it, it's in Fish and Fisheries. But really the idea here was by using SPM, we had this really dynamic, high resolution operating model, which is shown on the right for 4 different time steps, where the distribution of the species really changed pretty drastically over time. And also, we had really heterogeneous fishing effort. The length comps were both sparse and, heterogeneous. We had, I think, 7 fleets operating at different spatial temporal scales. Movement was driven by preference functions for SST, chlorophyll, and distance between cells. And I think for this talk, what I was really interested in was we did simulate the same kind of tag design that is available for yellowfin tuna. So our releases matched what was available, which I think was 2 or 3 releases over a couple years that are really located kind of near shore for immature fish. And so that I think it was interesting in that it kind of simulated these kind of limitations that we see for real world tagging data. And so it really led to a lot of issues. I mean, a lot of time was spent by the analyst groups kind of figuring out how to use the tagging data and whether they should use it. And generally speaking, it's kind of a mixed bag. A couple of groups decided that, it wasn't representative data. There's not much they could do with it. One did a spatial model, just didn't estimate movement. One did a spatially implicit model to avoid using the tagging data. But yeah, a lot of time was spent on how do you deal with tag mixing and all these issues with the representativeness of the tagging data. And so we did one post hoc analysis that I did just want to point out. And so basically the gray line is a 4-region spatial model, just without using the tagging data. And then the dark blue line is kind of the base spatial model that one of the groups developed. And so even with all these issues with the tagging data, the model was genuinely better off when the tagging data was included, even though there were issues with tag mixing and representativeness. I don't think you can take this finding and apply it to all kind of situations, but I think it's an interesting aspect of using tagging data and how powerful it is for estimating kind of movement and mortality, even with some of the limitations. And I do think these high resolution models really kind of made us realize that maybe we don't understand our assessment models as well as we think we do, based on kind of most of our simulations where the structure kind of matches. So I think there's some real power in kind of using these high resolution models to test our assessments. And so just to kind of wrap this up, we had another paper as part of that experiment that summarized kind of what spatial model platforms exist and use this to kind of summarize some issues that might be worth considering for future spatial models, which might be of interest here. And really for this, I mean, basically what we concluded was kind of the obvious, was that we need the ability to have multiple types of tagging data incorporated, have a wide flexibility in how you treat things like tag mixing and nuisance parameters. And yeah, to kind of wrap up for recommendations for kind of a next generation tuna assessment, I think what we have now, which is spatially stratified with tagging submodels, a lot of flexibility for how you handle nuisance parameters, tag mixing, and then also I think integrating what Jim and Matt were talking about with these these kind of highly flexible movement parameterizations that reduce the number of estimated parameters. So kind of linking all that is probably where what we need to have in the next one. I think it's increasingly necessary that we're able to kind of utilize all these new types of tags, especially electronic tags. Obviously, we'll talk about CKMR in future presentations. So I think those are the two main things. I think it is potentially desirable to kind of have this multi-scalar approach where you have the high resolution tagging movement that is directly integrated into the spatially stratified assessment. I just don't know how feasible that is in an operational assessment, how much extra time it would take. And so a couple discussion questions just to wrap up. I always come back to this first question, which is whether the tagging data violates assumptions more severely than other data sources, or if it's just really easier to identify, and so we kind of latch onto it. And then, I guess I still wonder, the high resolution models are really powerful, and I just wonder if we're getting better movement estimates out of them, or kind of Matt mentioned, when you start scaling up, maybe they come out kind of the same. So it'd be interesting to start looking into those types of things to see if we're actually getting new insight or they kind of come out in the wash. And I do think it's important, I think Mark brought this up, to maybe take a step back and ask, do we need more complex assessments to meet the goals of management? Or, is what we have kind of currently adequate? We just need to actually listen to the advice sometimes. And also, maybe we can start using these high resolution models to start doing MSE type analysis of how to optimize our data collection. So yeah, I think the other things are kind of basic to assessments, like how do we deal with diagnostics for tagging? How do we deal with data weighting? Things like that. And so yeah, I'm just acknowledge my co-authors again, and also that Matt has a pretty fun model he's been developing that's kind of putting the spatial aspect at the forefront in the development. We haven't used it for tuna or anything like that, but we've done it for sablefish with the tagging data. Thanks.
Okay, thanks, Dan. Does anyone want to start off with a question? So Dan, you've got a question here from someone. Let's see. David, do you have a question? Actually, we have to make you. All right. There we go.
Can you hear me now?
It's very low level.
Okay, hold on. Can you hear me now?
It's still quite low.
Quite low. Okay. Let's see if I can get
That's
audio.
That's good.
Change. How about now?
Yep, it's good.
Okay. Thanks, Daniel. I think you've actually brought a couple of the things that I wanted to ask because I think a central question to the need for developing this new model is, do we believe that it will reduce the uncertainty of stock status determination? And you seem to be suggesting that adding tagging data in spite of its limitations would improve the stock status determination. The second thing, which actually you also alluded to, is that maybe by having more spatially explicit assessment models that incorporate all these other kinds of data, we're going to be able to produce advice, management advice about spatially explicit management practices, which generally we don't for the most part. But I was wondering, and I think that's a little bit of a danger that I see, is whether really what's behind this is that as scientists, we are convinced that more realistic models perform better and are more trustworthy. And I don't know whether other stakeholders feel that way. Or even worse, that we are just pushing this because we need to justify the use of all these expensive data that we're collecting that are spatially explicit and that we are currently not using. So thank you.
Yeah, I mean, it's definitely a complex question. I think part of it is, the management, what you're trying to manage. Do you need to avoid, kind of localized depletion? If you have a single population unit, do you really need to worry about how they're distributed, or if the population goes lower in one area and not the other? So I think it's first a management question. And I guess the other part of it was kind of, do we need more complex? I don't know if I'm arguing for either more complex or less complex. I think with tunas, accounting for that spatial structure is probably pretty important. And going back to that simulation experiment was really interesting. I didn't present on this, but what we found is that doing a single panmictic assessment across the entire Indian Ocean for the yellowfin tuna versus breaking it into 4 regions and doing spatial assessment with movement or without, however the analyst groups structured it, didn't necessarily lead to more accurate, less biased either way. The only trend we saw across the analyst groups was that the spatial models always had a higher estimate of total biomass than the panmictic models. But some panmictic models were more or less biased than some of the spatial models, and some of the spatial models were less biased than some of the panmictic ones. It just depended on the analyst group and some of the assumptions they made. And so that's kind of my point, is I don't know that we fully understand our assessments as well as we think we do. And when we start doing these more high resolution data simulators and applying realistic assessment situations, I think we can learn a lot. And so, yeah, I don't think I would argue for more or less complex. I think we need to just do more kind of testing of them, try to understand, especially, context dependent.
Do you have a follow-up on that, David?
And no, thank you. I
Okay.
think I appreciate Daniel's answer.
Yeah. I just want to make one comment towards that. Is one of the big problems we have with our tuna assessments, particularly the tropical tunas, is trying to estimate absolute abundance. And a lot of our information on absolute abundance is coming from link frequency data, which is very problematic due to a lot of reasons, but also because of spatial differences in growth rates or whatever's doing it. And so tagging data is our best bet, but our tagging data is really bad because of the issues with trying to tag, fish representatively over the whole area. And so the spatial aspects come in to be important there. Jim, do you have a question?
Yeah, just 2 parts. Can you hear me okay?
Yeah.
Yeah, and thanks for putting this slide back up. Well, my first thing is just in general for tuna assessments. I think they're really different questions for temperate species, thinking all the bluefin, the need for tagging data and application seems like it would be much more straightforward in the sense that you pretty much know where the spawning occurs and you've got relatively direct things. With tropical tunas, I think the questions are really different, or the potential is quite different and frankly more difficult to actually be useful in the sense of this slide where it says management advice, in that if you, I'm just skeptical that even with the best model, our management advice could predict the relative abundance across regions in the sense that we could provide it. It's almost like predicting from a multi-species model that if we remove one predator, we're definitely going to get more prey of one species. That type of higher order problem, and I think it's going to be a long stretch. But I just wondered if it's clear at the outset of this workshop that dealing with tropical tuna and tagging and movement seems very different than temperate species. And then just if the goal is to really give advice, kind of to David's point, at a small scale level, what are the chances that it'll be good advice? And how do you test that?
Dan, do you want to respond?
Yeah, I mean, I think that's kind of like the last question. I don't know that I have an answer to that. I would again say that I think using these high resolution like CPODIME and models like that to kind of test some of the full management procedure, take the MSE approach and kind of use these high resolution models as the operating model. And then, testing, if you do a pan-MICTIC assessment, if you do CKMR or something, and being realistic about the data availability and also, where it's coming from across space and time. I think that is a potentially powerful way to start understanding what might work. But yeah.
Well, thanks, Dan. I think we'll move on to the next presentation. And the next presentation is by Tobias. Are you online, Tobias?
Can you hear me okay?
Yes, we can, yeah.
Nice. Sharing my screen. Can you see my screen?
Yeah, that's good. We can see it in full screen too. So.
Nice. Yeah. So my name is Tobias Mildenberger. Thanks for having me. I'm very excited to have the chance to talk to you. And the title of my talk is Go with the Flow, Adding Movement to Tuna Stock Assessments with ADMove. And this was my postdoc project a few years ago, and a big shout out to Mark and Anders, without whom this would not have been possible. The main motivation of my project, or our project, was basically the question if we can estimate quantities relevant for tuna stock assessments from tagging data, and those quantities being fishing mortality or relative and absolute biomass indices. And in order to estimate that from tagging data, it became very clear, well, we need to estimate the movement rates first. So we were working on this advection diffusion state-based model for tagging data, which I refer to as ADMove. And what ADMove can do, it kind of can combine conventional archival tags and can link movement to habitat preference function of environmental covariates. It can also incorporate spatial effort and catch information if that is available, and can then estimate length-based mortality rates and biomass indices. Throughout this talk, I will refer back to the case study of skipjack tuna in the Eastern Pacific Ocean. So this little guy will indicate when I'm talking about this case study. So thanks to Daniel's great presentation, we know about these different tagging types. And as Daniel said, and that's also the small insight I got into the tuna world, conventional tags play a big role. So these non-electronic tags where we usually have a release location, and for some percentage of the release tags, also a recapture location. And these are often like T-bars or spaghetti tags, as shown here. And this was also the tagging type that was available for our skipjack tuna in the Eastern Pacific Ocean from two main tagging programs. And we had almost 10,000 tags, of which 2,000 were recaptured. And that's also indicated here, blue dots being the release locations, and then the arrows indicating the distance to the recapture location. And they were submerged anywhere between 10 hours and 3.5 years. Another important tag, as also Daniel mentioned earlier, are archival tags, so electronic tags that record internal or external data, and they must be physically recovered. The image shows an example that is used in the Eastern Pacific Ocean for Big Eye, the bigger one, and the smaller one for skipjack. And here we have a little bit more information, so we also have a release location, but then we actually have this predicted track until the final recapture location. And this tag reconstruction or track reconstruction is based on the light measurements and pressure or temperature. So it's a model-based prediction. And we also had a few of those available from the latest tagging program for our skipjack. We had around 50, indicated again blue the release, and then orange the recapture, and the gray track the reconstructed track between the two. And they were submerged anywhere between 2 days and 8 months. So that was kind of the data that we had available. And if you then think about how to model that, after Jim's great talk, we are experts in advection diffusion models, but if we stick on the individual level first, then we could describe the movement in space over time by a continuous time-biased random walk, which can be approximated by this stochastic differential equation, which contains some deterministic movement part in x and y directions, and then some random noise here with a Wiener process and the diffusion matrix controlling the random spread. So with that, we can describe kind of the track of an individual. If we do that on the population level, then we arrive at this advection diffusion equation that also Jim and Ina showed earlier. And this is a very simple version of it because as it's written here, it would imply diffusion capital D and small u and v, the advection rates are kind of independent of space and time. That's of course not the case. So they depend spatially and temporally, and if you wanted to estimate that, you quickly end up with thousands of parameters for your crit size. So a nice way to actually estimate those parameters differently is by the use of the habitat preference function, and also that has been mentioned before. These are also part of Zepodim, and Jim also introduced them. But maybe I just reiterate them because they were also part of our solution. So given some environmental field, and it might be temperature, it might be some indication of prey abundances, if that's available, and some assumption about the habitat preference function, so which values of this covariate field your species might prefer or which they might tolerate, you can then basically convert that into a spatial habitat preference where dark red indicates areas of high preference. So here, for example, intermediate temperatures. And then movement would be informed by the gradient of that habitat preference field and be towards those areas of high habitat preference. And that's a pretty neat way because we can estimate those habitat preference functions as smooth functions, and just by using a few parameters instead of 1,000, but also as Ina said earlier, we are kind of assuming that we know or can figure out which environmental fields actually driving the taxus component, and that those fields are available. And then Thorsten, in the paper he presented this elegant approach to estimate. You still need to estimate those parameters of the splines or your habitat preference function, and this can be done by discretizing the advection diffusion PDE on a regular grid by the matrix exponential approach, using this generator matrix as a sum of your taxus component, maybe a passive advection due to currents, and the diffusion component. And we thought this was a really elegant way, and this was kind of our starting point. But when applying it to the skipjack tuna, we quickly run into the limitation that we are dealing with a huge area, all of the Eastern Pacific Ocean, and often quite strong advection. So there's a constraint about the crit size. If you have, like, the crit size has to be linked to the advection and diffusion. So you quickly run into long run times and large matrices of which you need to take the matrix exponential. So we were looking for alternatives and found some approximation with the Kalman filter, which now uses continuous space but discretizes time. And if we call back into memory the conventional tags and archival tags, we kind of can interpret conventional tags similar to archival tags, only assuming that all these intermediate steps are unknown, so are kind of latent positions, and we can then define a state-space model with an observation equation. Here assuming a multivariate normal distribution with some covariance, observation covariance, and we can also define a process equation where the position at the next time step is kind of dependent on the current position, plus some taxus and advection, and the uncertainty of that process scales with diffusion. And this is, of course, an approximation, assuming linearity and normal distribution, but we thought yeah, we give it a try. And so the question was, does it work? And we started very simple with a very simple simulation setting where we have one environmental field here in A, and then simulating some preference habitat function in B that would determine the movement, and we can release a bunch of conventional tags and some archival tags, and then just try to re-estimate that with both approaches. And for one replicate, it looks, for example, like this, where we can actually recapture or recover the estimates quite well, and also this habitat preference function. Note that we were giving the right environmental fields to the estimation model. You can do that, scale that up a bit and do that for many replicates, and you might see something like this, where you have the relative error on the y-axis and the parameters on the x-axis, and then the columns are the number of tags here for the matrix exponential. And of course, so we didn't see a bias, but the uncertainty, of course, depends on the number of tags that are available. The rows are then just different scenarios of only conventional, archival, or both tags together. And we saw a similar pattern for the Kalman filter. Again, no bias, but large uncertainty if you're dealing only with a handful of archival tags and 20 conventional tags, which is a very limiting case, I guess. But in addition to all of this, we also saw that the matrix exponential approach is actually dependent on the crit size. So if you scale it up to very large crits, you again get very uncertain estimates, and at least for the uncertainty of the archival tracks, also some bias. So that was enough for us to see it should work in principle and also in a small simulation study. So we applied this also to our skip check tuner and could see some preference for sea surface temperature and also the kinetic energy of the water. And this is kind of shown in A here. Some intermediate temperatures showed the largest preference, and high kinetic energy of the water showed the biggest preference as well. In this scenario, we set the diffusion constant in time and space. The results vary a little bit if you allow diffusion to vary as a function of environmental fields, or if you assume they are dependent, and also if you use either one separate of those environmental fields. But overall, from what we saw, the recapture location predictions and also the movement, it's relatively robust. There's always a question how to best visualize advection or the movement, and you could do it by arrows, but of course this varies over seasons and times depending on the environmental fields that you use and whether you model seasonally varying habitat preference functions. So that's tricky. Those were the results from the time. Since then, we were kind of moving the model from TMB to RTMB, and with a really fruitful workshop with Inna, Joe, Arnie, and Mark, we were also, we ended up, it was recommended to use not the kinetic energy of the water to inform taxes, but actually similar to Sepotim, the currents actually being a physical force on all individuals, so they might have to swim against them. So this is, for example, the estimated preference for sea surface temperature with the matrix exponential when including currents and with the new RTMB AD move package. So quite consistent results, at least for temperature when including currents, and quite similar results also with the Kalman filter. And then another way how to display the movement, and this is also how Jim showed it earlier, you could simulate or release a bunch of tags somewhere here by the small black dot. And then here, using the matrix exponential approach, you could follow the distribution of these releases over time in space. And this is the solution from the matrix exponential approach. To do something similar for the Kalman filter, you actually need to simulate them, and then using some bivariate kernel estimator to get this density in space. just to show there's some difference, but overall also some agreement, a little bit of movement, a lot of diffusion. Yeah, but this we are still kind of working on this. Not a lot of time left, but still a lot to say. This is kind of how you would set or could set up this generator matrix for the matrix exponential approach. So by using the neighboring cells, you could construct this instantaneous matrix. And actually, there's a really cool way to, well, a really elegant way to account for mortality. So Matt raised earlier the question, first movement, then mortality, or the other way around. Actually, in matrix exponential, you can do it at the same time. So individuals, they either move to another cell or they are being caught by any fleet. You could have several fleets in here, or they die naturally. So I think that's a really handy way in the matrix exponential to account for mortality. If you were to want to do something similar for the Kalman filter, that's a little bit more tricky maybe, but you could kind of account for each prediction at each location at the fishing mortality and the survival probability at that step. And you could do that in that way, so accounting for survival and then the probability of being caught in the end when they are recaptured. And interestingly, when you do that, also the non-recaptured tags find their way into the likelihood because they actually found some track throughout the space where they never were caught by a fishing mortality, by a fishing fleet. But this approach uses effort data, and here we lost a lot of hair trying to figure out how to depend less on this effort data, as yeah, this is really questionable in a Tuna world. And we looked at a lot of relationships, and now I'm running over time, but maybe just if you allow 2 more slides. So if you were to find a way to trust this effort data, you can actually estimate natural mortality for different length classes, and those estimates we estimated were actually quite in line with other studies. You can also estimate fishing mortality by length and fishing mortality for different fleets over time. And you can play that a little bit further even by plugging everything into the Baranov-Ketch equation and then actually having the only unknown in this equation would then be the abundance. So you have some abundance for each fine spatial crit cell, which you then can smoothen out somehow and get some biomass estimate. And what we saw is that, well, your fishing mortality effort assumptions determine the absolute scale of this, but the relative pattern is pretty consistent if you focus here on the colorful lines. Yes. And this was what found its way into the benchmark assessment. Yeah, there's not enough time to talk about the Peterson style method that we also worked on, but we have a long list of further to-dos, so we want to make all of this available to people interested playing around with that, and we're working on that. This linearity assumption of the Kalman filter is bugging us a little bit, so we're working on an uncentered and extended Kalman filter. The boundary conditions are elegantly solved for in the matrix exponential, but a little bit harder to work with in the Kalman filter. Residuals are difficult in those contexts, but very important in diagnostic tests, so we're working on those as well. Effort, yeah, we would like not to depend on that, so that is also work for further exploration. Yeah, and we will also start working on using this model for acoustic tags, which I think is quite exciting. So yeah, thank you for listening, and thanks for a bunch of people being part in this process.
Thanks, Tobias. I guess we got time for a quick question if anyone has one. Okay, well, we can always ask questions in the discussion section later on in the afternoon. Simon, are you there?
Yes, I am. Sorry about that. I'm working off a laptop and everything's a little bit difficult. I was just trying to get my presentation up. How are you?
Good. Yeah, we can hear you clearly. That's good.
Yeah, I'm running off 4G, so let's see how we go. I'll just share my screen.
Okay. Yep, we can see it full screen. That's good.
Awesome. Well, Mark, thanks for inviting me to the world of tuna to give my perspective from an invertebrate side. I think things are a little bit easier for us, and so we'll just start off. I guess a lot of the models in West Australia that we're using anyway, fleets are used to replicate areas or to solve the issue of areas, and therefore the issue of any movement between areas and so forth. It does require that, you know, over the breadth of your fishery, you're using parameters that are generally averaged, say average growth across the whole area. But, you know, this concept is so much faster and simpler to code and model. It's great. So it's obviously where we wanted to originally start with the lobster, just using fleets. So just a little bit of background into the Western rock lobster for us in Australia here, and I'm just looking at this part of the coast. This is our largest fishery, wild capture, single species fishery, but you know, it's relatively small on global standards. And so essentially this is the area over which we run our stock assessment model. And it's just into the biology to give you an idea about why we went down the road we went. If we, start down the bottom here with the larvae and the eggs, there's sort of a 9 to 11 months. They settle in the near shore, you know, less than 5 meters generally habitat, and they spend 3.5 years living in that area growing. And then it's this phase, the migration of the lobster, which moves the majority of these juveniles offshore into the adult breeding grounds, where a year later they mature and where they remain. So I guess the key points are, at this point, the lobsters do not move. They only move to forage up to sort of 500 meters. At this stage, we get lobsters living in the same den for over 10 years, 15 years. They move very, very little. It's only this stage in which we get substantial movement. It's one age class, and it only occurs for a period of about 3 months of a year. We've done a lot of tagging over the years. We've used electromagnetic trackers, archival tags, acoustic tags, but most of our tagging is the T-bar anchor tag, dorsally and placed ventrally as well. And as you can see, we've done over 500,000 tags released. We've got about 10% back over the journey, and the full size range of animals has been tagged and over a lot of years. And this is the results of a lot of those are the T-bar anchor tags. What I've done here is that we haven't actually tagged exactly at these spots. These animals within each of these regions its sort of release area has been adjusted to a common point for each area, just so you can see the pattern easily. And what stands out quite obviously is this pattern. We get, irrespective of where a lobster is really moving off the coast, they sit on about the same bearing, almost 283 degrees everywhere that they leave from. But once they hit this deeper water area, between 100 and 200 meters depth, they swing northwards into the predominant Lewin current and walk into the current. And this is a contranitant migration in which, you know, the bulk of the breeding stock sort of lives in this area. the eggs are released and the larvae move slightly southwards, come back in, and then they have to the juveniles have to move back northwards to maintain their position on the shelf. So with this sort of movement, you can see that the fishery is very interactive with other parts, and these are our management zones that we have in lobster. So we've got A zone, B zone, which is this whole area, and then C zone to the south. Now, all of our reporting for our harvest strategy is by management zone. So we need to produce estimates of biomass, egg production, and harvest rate by these zones. Now, in a simple scenario, we would therefore just model, say, zone C on its own, zone B on its own, or zone A. But as you can see, we get a lot of movement from zone C into zone B and A, from B into A, and then back into B, and so forth. So what's happening in any one zone affects what flows into a subsequent zone. So unfortunately for us, we have to model this fishery as a whole with the areas in there. So that's sort of the first tick why we can't just stick with fleets. The other interesting thing about this coast, it's sort of runs very much north-south along a dominant current, the Lewin Current, which flows from the north towards the south, bringing low nutrient warm water with it. And this results in the habitat changing dramatically throughout the fishery. The tropics are just here, the line of tropics. So we're very coral dominated to the north. We move through sponge domination and then down into algae and reef ledges to the south. So the habitat and therefore the biology changes dramatically up and down this coast. For example, some of our work from using the tags, we show that we get quite different growth rates depending on where you are in the fishery. This is offshore at the Abrolhos Islands versus the coast, and we get much larger animals on the coast than the islands. And this is quite extreme from north to south as well, with much larger animals to the south than the north. I guess another way of almost looking at that growth is the size and maturity. So here's some mature females from different parts of the fishery. And what we have, Fremantle is the most southern location we have this data for. And you can see the size and maturity for females at Fremantle is nearly 90 millimetres. And then offshore at the Abrolhos Islands to the north, it's 65. So dramatic difference in the size and maturity, which is a bit of a good proxy for juvenile growth rate towards this life stage. And we also have different natural mortality throughout the fishery. So I've just put two extremes up here. Some brownie tag capture work we've done in closed areas and adjacent to closed areas to the south of the fishery. Give us estimates of M of around about 0.12, whereas it's over double that in the north. Interestingly, this is a very sponge-dominated area with little good habitat for lobsters, whereas down here we've got much better reef and ledges. So again, this complexity of the actual fishery and the life history result requires us to end up with a fairly complex spatial model to try and account for all of these various aspects. This is the model we're currently running. I'll get to a new model that we're developing soon, but it's got 11 areas, which is ridiculously complex. 8 time steps to try and replicate some of this migration behaviour in its own time step. First the offshore and then the northern, but also other aspects of the life history of the lobsters. The females must be returned when they have a mating spermatophore or eggs. So there's certain time steps in the year where the mature breeding stock is protected and so forth, so they need their own time steps. And we, as I've got here, we've got catchability and natural mortality not only changes spatially, but between the migration phase, which is the whites, because they're a white lobster, we just refer to them as that, and the reds phase, which is the sedentary lobsters. So how do we actually go about moving them in the model and fitting to the data? As I mentioned, we've got two models that we're running with at the moment. We've got Lob 11. This is an 11 area bespoke model that was developed a number of years ago. It's a length-based model written in ADMB, and we move the animals by size. And it's a simple way to do it. You approximate the age cohort by doing this, but the problem is it results in multiple animals being able to migrate multiple times. If they're at the smaller size of that size cohort, when you move them, they can then go the next year, and it requires a few more parameters. In a newer model that we've been working on, Andre Hunt's been helping a lot with the development of this model, IMALT, integrated model using length transition. This model is in TMB. It's a length-based model, but it also keeps track of the ages of animals, the cohorts. So the big advantage of that for us is we can actually just move the 4-year-old lobsters, which are the lobsters that are moving. We don't have to worry about the size. It's a much better way to go about things. It limits the movement to only once. You can only be that age once. Less parameterization. The one hiccup with it is, and this is where I'm spending a bit of time at the moment, is how do you put in your tagged recapture data when you don't know the age of the animal when it was tagged or recaptured? There's a lot of similarities between both models, which I was just going to sort of tick through how they're working in the current model more so. In that we have a fairly simple movement migration matrix now. From Tobias's previous talk, you got dispersion and things driving the animals to move. This is, as I started off showing before, we've got a relatively simple situation here where we know in November and early December they're going to migrate west at about 280 degrees. We know that later on in December they will get deep enough and they'll swing north and spend the next sort of 2 months walking northwards. So we can designate the direction of which the lobsters will move and when, using a simple matrix and using time steps to replicate when that matrix is applied. We start, I guess the important concept for us was we start jumping through the matrix and moving the lobsters between areas in the south, so that some migration can maybe move you up towards Landsland, but then when we roll the movement through Landsland, Jurien, Dongara this way, an individual animal can actually bounce all the way up north, which does happen in reality. So it was important for us to roll the migration in a certain order, if you will, and it's relatively simple. We're fitting a normal distribution, if you will, to with a proportion moving from each area to its adjacent area, and then the numbers of that sex, size in that area. I jumped from one area to another and then removed from their previous area. The important thing for us with so many tag recaptures was that we put similar animals together. For example, all the lobsters of the same sex and same size released in the same area in the same month are pulled together, and their life history throughout the model is replayed collectively, so that they can we can say running through each animal each time, and then just at the point that each animal in that group is recaptured, we can have a look at where the probability of where it is within the model, and use that in the likelihood. In this model, Blob 11, and in IMOL, we're not applying Z to the released animals. In another model we've done, we were applying Z because we were trying to use the tag capture data to actually estimate biomass, and it worked out quite well, but it's a lot more complex. And in these two models I'm presenting here, our focus was more on using the tag capture data to inform the model of movement, and not so much in biomass, for simplicity reasons, essentially. And the important thing that we discovered when we're going through this was the requirement to weight the recaptures by the efforts in those areas. So we're quite lucky in that we have quite fine scale data records. The fishermen return daily records on a 10 by 10 nautical mile block. It's a pot fishery, so we get very relatively good effort information. So that's built in to weight the probability of an animal being observed and ending up in an area when it was recaptured. And then we're running through the observe with the probability and a multinomial likelihood. This is sort of an example, I guess, of one of the diagnostics we use to look at how well the model's fitting. This is lobsters released in Area 5, which is in the middle of the fishery, shallow water area, and where the observed and estimated recaptures have occurred. So this is released in Area 5. You can see the majority of the animals have been recaptured in Area 5. So either they were captured before they migrated, or they did not migrate far enough to leave that area. But the area directly offshore, Area 6, there's also quite a few recaptures occurred there. And then 10 is further north and offshore again, 8 and so forth. So it's a good, nice way to look at it. The couple of issues we have come up against is number one is the reporting rate. So we conducted some trials, we call them salting trials, and where we tagged lobsters and placed them in pots ourselves, and then sealed the lobster pot up with a device that when the commercial fisherman came along and pulled his gear, would fall off, so he didn't know that lobsters had been trapped in. Because our pots, they don't trap the lobsters; they just retain them. So we had to seal them in, and this provided us the ability to look across the fishery at the reporting rates that various areas provide, because that heavily weights where we were seeing our lobsters. And it was very interesting that we had one area towards the south in which we got very poor reporting rates. So it was valuable work to conduct this, to then build into the analysis to adjust the observed recaptures. The other difficulty, I mentioned unknown age of tags before. That's an issue we've got in the current modelling. But another issue that we've come up against, and one reason why we stick with these length-based models that use size transition matrices to grow the lobsters, is that I started the presentation off by showing how much the biology varies throughout the fishery. And when you're growing animals using growth curves, like in SS3, as an animal gets to a faster grower region, its age length no longer lines up with the growth curve, so it makes it very difficult to use them to change growth of the animal as it moves through the fishery, which is quite important for us in lobsters to account for that. So that's the reason we stick with length bins, size transition matrices, and tracking age through that.
Hey, Simon, I just want to note that you've run out of time, so try and wrap it up quickly.
All right. Well, that's me done.
Great.
That was my last slide, Mark.
Okay, thanks for that.
It was just good timing.
Yeah. So, okay, we'll open up to questions for Simon, but also any questions for the other presenters or just discussion questions in general.
The camera is super active.
Yeah. I mean, Simon's sort of points earlier on the issues with different sizes in different areas or different growth rates. And so one question I have is, why don't we just go to a fully length-based model for tuna, particularly since we don't have very good growth data, or not necessarily growth data, but we don't have a lot of age information, like age composition data. So would there be any disadvantage of using a length-based model rather than a age-based model, particularly if we're modeling tagging data and spatial models? Mark, you have a comment?
Yeah, I'll just fly the close kin flag at this point. For close kin, age and length both really matter, and you can't do it entirely in a length-based setting. Also, epigenetic age will eventually work to some level for tuna. It doesn't necessarily yet, but it does work for other fish, and unfortunately not lobsters, not rock lobsters, but that is going to help. So you may not be ageless forever, but if you went fully length based, you'd really bugger up your close kin chances apart from anything else. Over.
Thanks, man. Simon?
Yeah, no, I mean, I agree with that, and that's why this new model that I've been working on with Andre is a fully length-based model, but it tracks everything with ages as well. So we've built it in such a way that you've got a plus age group, if you will. We only track the ages of the lobsters for the first 4 years, and then we pile all the ages together after that. But it sort of gives us the best of both worlds.
So Nick, do you have a comment?
Yes, thanks very much, Mark. Yeah, I'll just wave the flag of the integrated age length structured model here. We'll probably cover this point a bit on day 3, but just to say that, yeah, retaining age aspects within the model structure, I think, is important, because some earlier work that we've done does indicate that there are age-specific effects in respect of length-based growth. Just having a length transition matrix means that you're applying the same probability distribution for the mean length growth increment across all ages within that length class. Now, that rules out the capability of modeling dwarfs and giants, where there is this age-specific effect going on within the length transition matrix. So that that's, I think, is quite an important reason to retain age structure within your length-based model. The other one is that it seems that particularly within the WCPO now, we are obtaining a lot more method-specific conditional age length data to help inform the estimates of growth. And that offers an opportunity to try to get some kind of information on region-specific differences in growth. So I think those are two reasonably good reasons to retain the age structure composition within the growth function. Thanks, Mark.
David, you have a comment?
Yes, thank you. Thank you so much, Simon, for that very interesting presentation. I was wondering, you mentioned that it allows you to basically estimate biomass by management area, and I was wondering, how do the managers use that information? And the second question is whether by using this spatially explicit assessment model, you can explain better any interannual variation in recruitment. Thank you.
David, so our harvest strategy for Western rock lobsters, it's a little bit unusual. We have the biomass, the female biomass, breeding stock biomass in four main areas of the fishery, each have their own threshold and limit at around sort of, I guess, 35 and 20% virgin. In addition, so I guess they're the primary indicators, and then the secondary indicator, if they're both achieved, is the harvest rate is then used in an MEY analysis by management area. So now down to 3 different areas. So we start with 4 with the egg production and biomass, and then we've got MEY for the 3 management areas, and that pertains to a much higher biomass, I guess, of sort of 55% roughly. It changes obviously with markets and beach prices, but and that evolution of that harvest strategy that way has a lot to do with people. You know, their investments in the industry are by management zone, but whereas the breeding stock is assessed more on a biological sensible scale in the certain areas. And so what was the second part of your question?
Whether the model performs better at explaining interannual variation in recruitment than a non-spatial model.
Yes, great question. So we are incredibly lucky with our fishery in that we have a recruitment index that started in 1968, and it's measured spatially up and down the coast. So at 9 locations every 28 days. And the ability for this, it's a settlement stage of the larvae, the purulus. And it's remarkable how well the purulus abundance can predict the abundance of legal lobsters entering the fishery 4 years later. So that data is obviously used in the integrated model, and by having the spatial complexity, it really allows us to fine-tune recruit, when there is a good recruitment to the north or south, and to reflect the subsequent biomass from that. It's interesting. This year, we're currently getting the best recruitment through that purulus index in the southern half of the fishery that we have had for 35 years. So that'll dramatically change the composition of the biomass in the model, and in the stock moving forward.
Thanks, Simon. Jim, did you have your hand up? I might accidentally put it down.
I did have it up, but it was just a clarification on using the term size transition. I think that's for length-based models, and if we're talking about age-structured models, converting to length frequencies, I think we should really be clear on the distinction of what that is versus what size transition is in a pure length-based model. So just it's a little bit confusing over the topics that you're going to cover even. And Nick, just I would say that in multifan and stock synthesis, generally we're talking about conversions. So there's a age to length conversion that happens, not a transition. Anyway, dumb thing.
Yes, Simon.
So Jim, were you talking about what I presented?
No, I thought it was just
Oh.
the language clarity from kind of Nick's comment on, age based and versus yours, which is really length transitions and growth length to length.
Thanks, Jim.
So has anyone got any questions? I've got a question for Ina. It's going back to the tagging model. I mean the spatial presentation, but there was tagging data in that model as well. Is your model kind of completely deterministic in the sense that everything's just a function of the environmental covariates, and so that's why you have very few parameters? So if the relationship with the environmental covariates isn't perfect, then the model is not able to sort of describe that discrepancy. And have you tried models where you've included more variation, like random variation or something else, to try and overcome that simplification?
Okay. Sorry, Mark. It's it's a bit it becomes quite late here, so if my answer will not be well done. Well, first, we are not modeling individuals, so we are not considering that complexity and stochasticity of individual movements. We are modeling the net movement of the large number of individuals. And yes, we are considering deterministic model, which, as you know, for example, diffusion part approximates the random movements of individuals. And also the individual movement toward the gradient includes also the random part, like the random turning and insertion of the habitat. So this part is covered, in my opinion, by the advection diffusion reaction model. For the environmental links, we have flexibility to test different mechanistic functions. Like, for example, for the tolerance to the temperature, we can use the simplest. We can start using the simplest Gaussian asymmetric function, asymmetric Gaussian, the logistic threshold, something that would allow us to estimate different shape of this relationship between the temperature or the prey abundance and the survival of larvae, for example, etc. So I think given that we have enough data to constrain those processes and to observe, to validate the estimated mechanism on the independent data, we can say that we do it quite well.
Yeah, thanks. I guess sorry to go on with this subject, but so in stock assessment models, we estimate annual recruitment, right? As a kind of like it's a random effect penalized thing, but it's really like a free parameter every year. But in your model, that's it's a emergent property of all the assumptions about the environmental variables and the dispersion and the mortality and stuff, right? So,
yeah, please.
So we don't. So if you run your model for 30 years, you don't have 30 recruitment parameters.
No.
But do you?
No.
I mean, I was just wondering what's the difference between these two, because you have a lot less parameters, so you have less flexibility. is there something that could be added to give it more flexibility, to be more similar to a stock assessment model in terms of the freedom of things like overall recruitment and things like that?
I think I would go with the suggestion made by Daniel that we need to test. We need to test our models and to compare them given the assumptions. I wouldn't take it as like we are making an assumption about making many assumptions in our model about the environment, because we are taking environment as it is modeled, the physical environment, which is assimilated, for example, the observations of the temperature and so forth. We assume that the environment is correct. We can have uncertainty, we should surely have uncertainty in our estimation of how this environment affects the recruitment, but we try to go as direct to the spatial distribution of larvae, for example, which is directly linked to the spatial distribution of spawning adults. Yeah. Okay, and then just add this variability, environmental variability that is also observed and experimentally established. So I think we need to test different models.
Any other questions? David?
Yeah, what the question you just asked was really interesting. I did not realize that those differences in recruitment. So I think in how you consider recruitment, one where is totally predictable, the other one is where we think that we actually can estimate, and the paradigm in fisheries has been that recruitment is very difficult to predict. So it's very interesting that these other models seems to not agree to that paradigm. I find it really good that we're exploring that possibility as well.
Anyone else got a question? So I guess following up on my questions to Ina, since I mean you know, we have two different types of modeling approaches. There's kind of the large box ones, which is the stock assessment ones, and then there's the fine scale ones. And CAPAM is probably the closest that we have to a full stock assessment using fine scale spatial processes. I think SPM is also, but I'm not sure if we have anyone on the call who's got familiarity with that. But I'm just wondering, how long does it take to run a CAPAM model? I mean, what's the computational demands for that model?
Okay. To run a simulation is very quick. Depends on the species and the domain considered. Example of skipjack, the Pacific Ocean 1 degree is a matter of minute for 20 years, 30 years. And we have simulations for starting 60 to 2022. The optimizations, the MLE are obtained on the shorter windows, like 20, 25 years. And those are usually one MLE runs 24 hours for skipjack. Given that we have enough the PCs with enough memory, we can run tens of those in parallel. So we can have a jitter of runs in parallel. But then because we explore different scenarios, different assumptions on our structural, on the model structure, which functional relationships we use, for example, whether we use the early larvae observation model or not, and so forth. We can have different setups of the optimizations. plus we have fixed parameters. We have different growth and maturity, which are fixed. We have some other parameters which are still fixed because it cannot be observed. And so you need to do this study of the optimization fixing over the grid, even though they are not observed from the data, but they still can change the outputs of the model. So the overall work with one forcing can take several months, but I think it is quite feasible given the problems we are all trying to solve when we are talking about like 3-4 months. But the great deal of time is spent on studying different resolutions and different forcing models.
There's a question in the messages from Daniel. Do you want to?
Yeah, I mean,
yeah,
it wasn't really a question. It was just kind of pointing out that I think there are ways that you can kind of bridge those two approaches. If you kind of started doing egg production versus estimating a stock recruit relationship and including the larval dispersal from age 0, I think you can kind of start to bridge the CPDIM and the stock assessment kind of approach, but I think there are ways to do it.
Daniel, you mean like an egg production type index that would be added?
Yeah, I mean, you can have fecundity. So how many eggs are being produced per spawner? If you have a spatial-ish model where you know where the spawners are, where they're kind of releasing, and then if you had information on dispersal, you could then simulate it the same way you kind of do with CPDIM. And then if you had like a larval index or something like that, you could fit that as data. And can't remember, there is like an SNA1 snapper model from like 2004 that kind of does all of that. It's kind of a research model from New Zealand.
May I comment on the using the larval data? We don't actually have a good data set for all tuna species. Like if we can consider a good abundance index of larvae for skipjack, and maybe assess substantial information on albacore spawning from the gonad data and from the larval sampling data. For yellowfin and big eye, we are not that rich in terms of data and constraining those processes. I think this should be considered.
Any questions? Yeah, Caroline.
Yeah. Okay. That's for Ina. Have you ever compared the estimated recruitments partition by region with the CPDIM?
In one of the slides I was showing comparison with multifacial, there was the upper plots was for recruitment. So the latest assessment are quite coherent in terms of the temporal trends of over all regions, although the differences between, if we are comparing them region-wise, are substantial. So we can have very bad correlations and discrepancies in terms of the estimated recruitment, absolute values. So, and in my opinion, I don't know how to explain those, but I have some ideas, but I don't want to impose them without doing proper. investigation.
Dan?
Yeah. Kind of on that point, I think it was kind of interesting because you showed that the Cipodyme biomass was always kind of higher than the multifan CL. I don't know if that was a spatial multifan or one area, but I guess I kind of mentioned in my presentation when we did the spatial experiment, we kind of had that same trend where the one area model, so the lowest resolution model within a given analyst group. So if they both did a single area model and say a 4 area model, the single area model always had a lower biomass. And so it was kind of interesting to see that where yours, Cibodyme, would be kind of the highest resolution model, and it was kind of higher in scale than the multifan model. And so it's an interesting thing that I hadn't seen before we did the experiment, and now seeing kind of a similar trend in yours, it does make me wonder if there is something going on where the spatial models have some sort of biomass floor scaling situation that kind of leads to that kind of higher scaling, and what's driving that.
Yeah, I tried to mention these things for raising it again, Daniel, but there are two reasons for that. There is a good reason and bad reason. So the bad reason is that it can be due to the 2-coarse resolution to represent a spatial phenomenon. And the spatial resolution can generate things like artificial diffusion in addition to being 2-coarse per se. The good reason is that we have the process-based model, process-based density predicted and not observed. This is, I think, that's advantage of such models. I don't know if it's clear. The process-based.
I think so. I'm just trying to kind of piece it all together between, say, a 1-region model, a 4-region model, and a 100-ish region model, and how they all kind of play together.
Yeah. Those comparisons are shown for against multifan CL with 6 regions, 8 regions. Sorry, so.
David.
Yeah, I understood that part of the reason was that Cipodyne is predicting biomass in places where we have no observations from tagging or from catches.
Exactly.
And therefore, that's why, this is predicting biomass on the basis of the forcing variables at finer scale. So maybe I'm completely wrong.
No, you're not completely wrong. It is linked to the forcing, of course, but it is the density. It is more linked to the process, like to the reproduction in the areas where the tunas are not observed, because the conditions for reproducing are also good, and the density of spawners are not 0. It can be predicted in unobserved areas due to movement and other process, because biomass migrated there, and fishers do not migrate or do not move with the biomass, because I don't know, during the ENSO tuna moving in the Central Pacific, and before there were no objects there were no fats, like on a large scale released fats. And so these tuna were not caught in the Pacific, and you look at the stock assessment at that times, they were estimated much lower stock. But in Cipodyme, we just how do you call it? You just run the model retrospectively, and you find out this stock in the Central Pacific. So that's why I say this process-based biomass, unobserved biomass, is a good thing of the mechanistic models.
So, Ina, what is not in Cepodime, which is in multifan or other, or maybe what you'd want to use in a stock assessment context, that you would have to include in Cepodime?
The uncertainty estimation is not as well developed yet as is in stock assessment currently. Second point that because of which I would advocate for not using CPDIM directly as a stock assessment as is that the dimensionality, and even if I said that it runs quite quickly, one day for convergence of the MLE for skipjack. If we go to yellowfin, big eye, albacore that lives for 20 years, it's longer. So we are working currently on making our serial application parallel. So I think it will move closer, this model for the stock assessment. It will be simpler to manipulate, faster to operate. And there's something else that I consider as a drawback of the model is that we don't have the weight dynamics, or it is not length based. The weight is not varying in space and time. We have it fixed. And we know, for example, for such species as big eye, we have substantial differences in the between Eastern and Western Pacific. And from the recent simulations with ICA-Moana, we actually saw that CPDIM has good potential for presenting it because with our habitat distributions, we can explain the differences in growth in different ocean basins.
But you're saying that currently
we don't have it. We don't have it currently in the model, but we can do it. It's still increasing the dimensionality, adding more equations to it. And we cannot do it right now because we are blocked by the computing power. That's why my suggestion would be to use it externally, to use it to collaborate and to use it as a testing model, to use it maybe in the harvest strategies for projections, climate projections, or I don't know, seasonal, decadal if possible, if the forcing is available.
Nick, do you have a question?
Yeah, thanks, Mark. Yeah, I'd just like to support Ina's suggestion there about improving the code to run in parallel, or at least multi-threading. Dave did some experiments with this quite a long time ago with parallelizing the tagging group calculations, because that's one area of the calculation where it's quite feasible to parallelize the calculations of the tagged component of the population. And he was getting pretty good improvements in the performance of around 40% or so with the tests that I ran. So yeah, I just look forward to that part of the discussion on day 3 in ways where we can multi-thread a lot of the internal model calculations that take place within one single model evaluation. So I think really support Ina's ventures into that area of developing CPDIM to try to achieve multi-threading of those model evaluation calculations. Thanks, Mark.
Yeah, thank you, Nick. Can I ask a quick question? Did Dave manage to make the estimation parallel or just a forward run of the model?
Yeah, no, he was just making the individual tag group mortality and movement calculations in parallel. Because, for example, with skipjack, you've got up to 270 individual release groups. So those calculations can be done in parallel.
Okay.
But the actual objective function calculation is done once all of that model predictions are available. Thanks, Ina.
Yeah, thank you.
David.
I want to support her suggestion on that we can, I think Cipodime could be very useful in the context of harvest strategies, because we often are looking for ways at developing reasonable hypotheses for robustness tests. I think that in many assessments that I've seen, the scale of the biomass is we have great uncertainty in the scale of the biomass, and I think Cipodime could provide a hypothesis about a potential scenario that would be useful as a harvest, as a robustness test in MSC.
Thanks.
Thank you, David, for this feedback.
Okay, anyone else got any questions? Jim.
Yeah, sorry, I missed part of the talk before, but I just wonder how much any fishery kind of summary data can be used to supplement kind of tagging, both tagging data and Cipodime results. Basically, are they consistent or not with what you would expect, kind of mean length or kind of modes changing over time? Is that used and is it something that could be easily brought in? I mean, spatial time steps, what the map of size as the fish were.
Okay, thank you, Jim, for your question. We have the parameter estimation in the full likelihood context, meaning that we have simultaneously 4 different terms, one for the fisheries catch data, catch or CPV, the length data, tagging data, and the early life data. So they are all minimized simultaneously, altogether as a sum. So we have a combined likelihood, and that's why, yes, we do have consistent estimations. We actually, the full population dynamics model parameters are learning from the movement estimated from the tagging data. And yes, tagging data, for example, can even impact even though in the tag observation model, we don't have mortality, natural mortality, we only observe movement and inform movement from tagging. But we know that the tagging data influence the estimation of mortality coefficients.
Cool. Thanks.
Okay, so it's nearly 3 o'clock, so I'm going to end it today. So thanks everyone for your presentations and the discussion. And we will be back tomorrow at the same time, 11 o'clock. And we'll start off with random effects in the morning and then move to close-kin marker capture in the afternoon. Thanks, everybody.
Interesting.